Yeah, I hope I'm audible.
Yes, sir.
Okay. So yesterday we had a very detailed discussion about uh, how we identify a suitable research question, how we go about doing the whole process of creating, asking the right question, such that the question has important components in it, the population, the intervention of interest, the comparator, the outcome. So we discussed at length about all those things. Then we looked at even how literature has to be, how to identify literature, how to do a thorough literature search using PubMed database. And then we also looked at what are the different kind of study designs in clinical research, particularly in the context of observational study designs. And then we had a very important discussion about the ethical aspects of uh, clinical research, particularly with respect to ICMR guidelines. Now, in the context of observational studies, we spoke about descriptive and analytical studies. So, so we have observational studies which could be either descriptive or analytical. And descriptive again, we have things like case reports and case series, or it could be a descriptive study that is cross-sectional in nature, more like a survey kind of design. And in descriptive studies, the important thing is you do not usually have a priori hypothesis. You do not usually have uh, you're not trying to find a relationship between an exposure variable or a risk factor variable and an outcome variable. You're just describing the population, and largely we're talking about incidence, prevalence, and such kind of determinants in your study. When we are talking about analytical study, we would be having a risk factor usually, a causality factor, and an outcome variable. Like for example, does smoking cause lung cancer? Okay, so this could again be subdivided into either cross-sectional or case control or cohort studies. So this is largely what we discussed yesterday, and we looked at how each of these designs, the cross-sectional has its own unique advantages, the case control has its own advantages, and the cohort studies also have their own benefits. So I think Dr. Palnivel has summarized it at length and how, in which situation, which context each of these designs are suitable. That has been discussed at length. So today what we're going to do is we're going to look at the experimental designs or the interventional studies. So when we're talking about word interventional studies, essentially we are trying to say here that these are studies where the investigator assigns the interventions to the participants. So the participants are receiving the intervention only because they are belonging to the study. If not for the study, they would not have been possibly receiving those interventions. And so they are all research participants who are receiving these interventions for the sake of the study. Okay, that is how you call it as a, that is when you call it as an interventional study. This intervention could be either a drug, or it could be a device, or it could be any a surgical technique. Okay, so it could be different forms of intervention. Now, the simplest form of intervention could be a single arm study, or it could be even a multiple arm study. So, what exactly is a single arm study? Now, in a single arm study, as the name suggests, it is just one group of participants that we have, and that one group of participants, all of them receive the same intervention. There is absolutely no difference. The outcome is usually the improvement from the baseline in the variable of interest. For instance, you have a situation like this. We have patients who are newly diagnosed hypertensive patients, and then you measure their systolic blood pressure at baseline or day zero, record it, and then give all of them, I mean, as after you explain to them about the study, take the consent, and then you give them the drug therapy with a new drug, say drug X, for a period of 12 weeks, that is 84 days. And at the end of 12 weeks, you ask them to come back, and then you again measure the systolic blood pressure, and then you find that what is the difference in systolic blood pressure at day 85 compared to day 0. So this improvement, is there a significant improvement in the systolic blood pressure? Has the systolic blood pressure come down after giving them this new drug for a period of 12 weeks? So this is what is called as a single arm study. So it sounds very simple and sounds very convenient. So the advantage of doing a study like this is that it is ethically, I mean, when you're talking to a patient about study, there is no question of an option there where they are going to be given different drugs. All of them are going to be given the same kind of drug. So it is something a little bit more easy to explain and convince the patient to participate in such a kind of a study. uh, because there is no one group may be an inferior or a superior kind of thing. So all of them are receiving the same kind of drug. And in terms of convenience, it's obviously simpler, easier to perform such a kind of experiment. So it's just a before after comparison. In statistical terms, probably you'll be doing a paired t-test and saying that there is a insignificant improvement in the blood pressure, reduction in the blood pressure. However, there is an obvious problem with these kind of studies. The problem is that how are we sure that this improvement in the blood pressure that you see at day 85, how sure are you that this improvement is because of the drug X? Maybe it is because of other factors, like maybe just the patient became very conscious of the fact that he has a high blood pressure, and he's started living a healthy lifestyle, maybe in terms of his diet, in terms of his exercise patterns. So there are any other factors, the n number of factors that could also influence the blood pressure. So one cannot really come to the conclusion that this improvement that we see in the blood pressure is because of study drug X. So there is a high risk of bias in this kind of study design. So it's not very ideal to have this kind of single arm studies, because it's very difficult to attribute the improvement as something that could be exclusively because of the drug X. So intuitively, we could say that having a control makes sense. You have a control group, that is a group of people who have not taken this drug, and you follow up them for the same period of time, and then you compare the difference between the two. Then you could probably say in more confident terms that one drug is superior to the other drug. So control group, having a control group, okay, it could be one group. So I mean, it could be two groups. So one is a study drug and one is a control drug. One group receives the new drug, one group receives the standard drug, and you compare the two groups. Okay, so you are having a comparator now, and you are able to compare the differences between the two groups with respect to the change in systolic blood pressure. For example, here you see eligible patients with hypertension. One group is receiving the new drug and the other group is receiving the standard drug. You measure the systolic blood pressure at baseline, and then you measure the systolic blood pressure at the end of treatment, that is after 84 days in both the groups. And then you compare this systolic blood pressure improvement that you see here when compared to this SBP change here. Is it significantly different? Is the difference here and the difference here, is it statistically different from each other? So when you do this kind of analysis, you are a little bit more convinced than the first situation where you have a single arm study, where you just had one group. So now you're more confident to say that it is group one, this new drug, which really is causing the difference in the blood pressure. And what do you call this kind of study design? This is called as a non-randomized controlled trial. Non-randomized controlled trial. Now, in this kind of study, everything is fine, but there is still one major problem, and that is with respect to how exactly the interventions were allocated to both the groups. Okay, so in a non-randomized controlled trial, you are just assigning one group to receive new drug and the other standard drug. There is no particular procedure that is being followed to decide who will get study drug and who will get the control drug. Okay, so that is a problem. Why is that a problem? Because it is possible that there could be some amount of selection bias when you choose who gets new drug, who gets the standard drug. For example, you could have a situation where you, as since for example, I'm an investigator, I want to prove that my this new drug is effective in this condition, in hypertension. So what I do, I take people who I feel are more likely to respond to the study drug, I give them this new drug, and to those people who I feel will not respond well to the study drug, new drug, I would give them a standard drug. Okay. So what is happening here? There is a subjective bias that is there on the basis of my presumptions, on the basis of my assumptions. Okay. So this is called as selection bias, and obviously this is going to have a huge impact on the outcome of the study. So it is not a very ideal situation to do such kind of trials. That is called a non-randomized controlled trial. So it makes sense only if, unless we remove the selection bias here, this would again become a very inferior kind of study at the end of the day, because you're not very sure the response that you see is because of the drug, or is because they are people who as such are highly sensitive people who will respond anyway to any drug. Okay, so you go for a better study design, and that would be a randomized controlled trial. So what exactly is a randomized controlled trial? In a randomized controlled trial, again, it's the same group of people, eligible patients with hypertension. You explain to them about the study, take the informed consent. But when you're taking the consent, you tell them very clearly, look, we are going to do this randomized control trial, and now you may be assigned either new drug or standard drug, and this is going to be decided by a random sequence, and that random, based on the random sequence, you may fall into this group or you may fall into the control group, that is a standard drug. And then you give them this new, you take the baseline measurements, take the follow-up, give them the drug for 84 days, and then take the again systolic blood pressure at the end of 84 days, compare the difference between this group, compare the difference between this group, and see whether this systolic BP difference between the two groups are significantly different from each other. So this is called as a randomized controlled clinical trial. Okay, randomized controlled trial. So how is this randomized controlled trial superior to the non-randomized controlled trial? It is superior because you are removing the obvious selection bias that would have otherwise occurred if you had not randomized patients. Okay. Now, what exactly is this randomization? Okay, we've been repeatedly saying randomization, randomization. What exactly is this randomization? Randomization ensures that every participant has an equal chance of getting allocated into either arm A or arm B. Okay, and why is this done? Primarily two reasons. One is it minimizes the selection bias that I was talking about. And number two, it could also minimize the known and unknown confounding factors. Confounding factors are basically other factors which could also influence the outcome apart from the variable of interest. See, we want to see whether it is the drug which is reducing the blood pressure. Okay, but there could be other factors which also influences blood pressure control. Maybe it is the BMI, maybe it is the patient's stress levels. So whatever XYZ parameters are there, which are possibly confounding factors, when you do a randomization has this ability to balance these factors between both the groups, so that at the end of the day, more or less, all factors are getting balanced between both the groups. Okay, so that is the magic of randomization. It minimizes confounding and it minimizes selection bias. Okay, so that is what is randomization. That is why randomization is superior than a study where there is no randomization done. Okay, so now next, how exactly do you randomize? How do you randomize? Now, the simple thing that you could do is you could do something like a coin tossing. Okay, you could throw a die, but even though coin tossing and throwing a dice are very valid methods of randomization, there is a problem with these kind of methods. And one of the major problems with these kind of methods is that you would not be having an audit trial for this kind of audit trail for this kind of methods. So when you're doing a clinical trial, there would be a lot of audits happening. So we would want to go back and revisit to see how randomization was done. So you could not really do a much on the going back to see how the randomization was done when you follow these kind of methods. So the standard methods that are these days employed are the one is the computer-generated randomization sequence and the age-old random number tables, which are almost not used these days with the advent of computers and software programs. So there is this software called random allocation software, which is freely available on the internet. So you can download this and you can see how it can be used, where you just have to feed what is your sample size, you have to feed what are the number of groups, you have to feed a little bit more additional data, which I'll be talking to you about subsequently. So now I have a question to you. An investigator plans to conduct a randomized controlled trial in patients with chronic obstructive pulmonary disease. Okay, so he wants to compare two drugs. One is roflumilast and the other is azithromycin. He wants to compare these two drugs. Among these two drugs, which is better to prevent exacerbations, that is worsening of the disease. So he uses a computer program to derive a random sequence and takes a printout of the same. He uses this list to randomize study participants in the hospital. Comment on the study. So do you think this kind of method of doing the randomization in this randomized control trial, do you think everything is fine or is there a problem? So you can probably think for a few seconds. On the surface of things, it looks as if this is a very valid kind of situation. It seems very valid because the reason is we are doing a randomized control trial. We are having a sequence also available, and the sequence is generated from a valid program. So everything seems right. But then the problem comes when you're having an investigator who has a complete sequence in his hand, and he is going to recruit patients from the outpatient department. So it is possible that now that he has a sequence in his hand, he could again conveniently choose who will receive roflumilast, who will receive azithromycin. He could decide, okay, this patient who is sitting in front of me, the sequence says he has to get roflumilast, but I know that this guy is not going to respond to roflumilast because that's my assumption. So I will not recruit him into the study. I will just allow him to pass, and I would take the next patient whom I feel will be likely to respond to roflumilast. So again, the same problem begins from the beginning, that is selection bias. So how do you remove the selection bias? You have to devise a method by which this random sequence the that is generated from the computer, that random sequence should not be with the investigator who's recruiting study subjects. So study participants are recruited by a person. That person should not have this random sequence. They should not have a knowledge of this sequence. So in other words, you should conceal the sequence from the investigator who is recruiting the participants in the trial. So that process of concealing the sequence, a randomization sequence from the investigator who's recruiting the participants is called as allocation concealment. So allocation and concealment is a method of concealing the randomization sequence from the investigator recruiting study participants. Now the next question, how do you go about doing this allocation concealment? Now there are some standard methods available which I just displayed here. One is the SNOWS method. Okay, what exactly is the SNOWS method? The SNOWS method is serially numbered opaque sealed envelopes. So you have serially numbered opaque sealed envelopes. So these are nothing but, suppose you have your sample size is 100, and in this 100, so you make 100 opaque envelopes, you number them from 1 to 100, and then you generate the sequence, and then you put a slip of paper into each envelope on the basis of the sequence. Like for example, if one is roflumilast, put a paper where it's written as roflumilast inside the envelope number 1. If sequence 2 says it should go to azithromycin, write azithromycin in a slip of paper and put it in envelope 2. So in this way, you do it from 1 to 100 and then seal the envelopes. So the envelope, it's important that the envelope is not a transparent envelope that can be seen through the light. So it should be an opaque envelope. So serially numbered, number it from 1 to 100 or 150 according to your sample size. Ensure it's opaque. Ensure it is sealed once you complete this process of putting the allocation sequence inside the respective envelope. So once this envelope is sealed, you have what do you have? 100 envelopes that are serially numbered and they are opaque and they are sealed. Okay, and this is given to the investigator who is going to recruit a participants in the study. So the investigator who's recruiting the participants, what they have to do is every time a patient is eligible, they take the consent, ask the patient whether he's willing to be part of the study, whether he's willing to be randomized. He signs the consent, and after all that is over, take the envelope number 1, open the envelope, remove the seal, open the envelope, and then according to whatever sequence it is, that particular drug is given to the patient who is part of the study. So this is called a serial numbered opaque sealed envelopes. The other method is, so you need to have someone who can do this for you. If you are an investigator in a clinical trial, you need to have someone else who can take care of this, making these envelopes and doing this whole process, and someone who can generate the sequence as well. So the other part is called as third party randomization. Third party randomization is, okay, you don't want to waste time sitting and making all these envelopes business, but you have a reliable person who can generate the sequence and who's always available on call, but who's not part of the study. So all you do is, whenever you are having a patient who's eligible, you can just call up the person, this third party person. Maybe it could be a pharmacist in the hospital. You call the person and say, this is patient number 2. Now please tell me what randomization should be, assignment should be given for patient number 2. And then he says, you have to give azithromycin, then you give azithromycin. The next day, patient number 3 comes, and then again you call up this person, and he tells you what is the allocation to be given. So this is very simple, third party randomization. So it's important that this person is not connected with the study. He's totally a neutral person who's not aware of anything that is happening in the study. So this is called as third party randomization. So these two methods are used mainly in more of trials which are single center or maybe just 2-3 centers, and it's more like an largely an academic setup. You kind of do these kind of methods. But in a commercial clinical trial where you're having a pharmaceutical company which is doing, obviously these kind of methods are not suitable. So you would be having better methods to do randomization. Usually they have their own computer websites which can do the randomization process. All that you do is you enter the data of the patient. There's some basic data they would ask you. Enter the age, the gender, the age, and the date when the patient is. So you enter all this, and then automatically it gives you the randomization sequence, the number, and you take that corresponding intervention and provide it to the patient. So this is interactive web response system. or interactive voice recording, voice response system. So IWRS or IVRS. Nowadays IVRS is very less used. It's more of interactive web response system. Then we have a different within randomization. Again, we have simple randomization, block randomization, and stratified randomization. Now, in simple randomization, what you do is you just randomize people into two groups. Okay, there is no other rules apart from telling what is the allocation ratio. Is it 1:1 or 2:1 or 3:1? Just that you have to tell the software which is randomizing, and it will give you a sequence. So you will find it. Let's, for example, here we have 100 patients. So there are two drugs given, roflumilast or placebo. So at the end of it, you have set 1:1 ratio. It would give a sequence in general, where at the end of the study, at the end of 100, you will be able to see that almost 50 people will be roflumilast and 50 people would be receiving placebo. But the problem with this simple randomization is that at some point you are stopping the study for some reason. Let's say you're doing an interim analysis, or even if you're doing a study where for some reason you couldn't complete the study, somewhere in midway it has stopped, then you would be finding that the groups are a little bit unequal in their distribution. Okay, so to eliminate this, we usually go for block randomization. So what is block randomization? It is a technique used to ensure that the subjects are randomized and equally distributed, but within blocks. Okay, so you randomize not the whole 100, but you randomize in small blocks. That small blocks you have to determine. Maybe it could be a block size of 6, maybe a block size of 10. Okay, whatever be the block size that you have to determine. So let's say if your block size is 10, then what you are trying to say is in every 10 blocks, for every 10 patients, there will be 5 receiving uh, roflumilast and 5 receiving placebo.
No, Sarkar.
Okay, so this is called as block randomization. So
I
you
did
can
not
see,
attending that. What was
please mute
it?
your audios, please. So, so we have this block randomization, as you can see here. This is the first block where there are 10 people out of 10. You would be seeing that 5 have received roflumilast and 5 have received placebo. In the second block, again, you would be seeing 5 placebo and 5 roflumilast. So this is called as block randomization with an allocation ratio of 1:1 and a block size of 10. So in order to make it little less predictable, you could have a variable block size. Instead of fixing it, okay, my block size is 10, you could have one block, maybe 10, another block, maybe 20, another block 30. So it could be variable block size also. It becomes very difficult for the investigator to predict what would be the next sequence. Then we have lastly the stratified randomization. What is a stratified randomization? It is a technique for ensuring that all variables that are having a very key or important influence on the outcome, that at least is evenly distributed between two groups. So you may ask me, you just said now randomization ensures all the factors are equalized between groups. But the problem is when you have trials which are little small, in spite of doing randomization, there is a possibility that one or two factors, important factors, might be imbalanced between the two groups. Okay, so you select which factor is most important that you want to balance between the two groups and ensure that those factors are at least evenly distributed between the two groups by doing a stratified randomization. So as you can see here, we have 1,000 patients here. So these patients are stratified based on gender and based on the age. Okay, so for this trial, age and gender are important. So accordingly, they are ensuring that more or less stratification is done so that at the end of the day, equal number of people receive both the drugs, but then there is not too much difference between the young and the old between the two groups. Now, so far I was talking about randomization. Okay, so randomization is mainly, remember, to prevent selection bias. Okay, so remember you could always do randomization in clinical trials, because randomization is all about generating a randomization sequence. Okay, but and but blinding is something different. So in blinding, what you do is you are trying to ensure that both the interventions in the study, you're having a study drug and a control drug, both the interventions look similar in its physical properties, the way it looks, that you cannot really distinguish the two in its external packaging as well as its appearance, so that you are able to prevent this assessment bias that can happen. Can you please mute your audios? We are hearing a lot of disturbance. So this blinding could be single blind or double blind. So when you say single double blind, essentially what we mean is the patient, that is a participant, as well as the investigator, both are blinded. Whereas when you say single blind, it's usually the participant who is blinded. You could also have a situation where investigator and participant are blinded, and one more person is additionally blinded, and that is the outcome assessor. So outcome assessor blinding is basically the person who is assessing the outcomes. Let's say someone is making telephone calls to find out whether the patients are doing well, whether they are having adverse events, whether they are having any things like someone who is measuring the echocardiography, or someone who is doing the ultrasound, someone who is making CT scan measurements. So all these people who are measuring important outcomes, which are actually going to determine the conclusion of the study, such a person is blinded. So he will not know what group a person belongs to. So that is called as blinding of the outcome assessor. In fact, many trials, it may not be possible to blind the investigator, it may not be possible to blind the participant, but at least you can blind the outcome assessor. It's much easier to do that. In fact, some trials you will find only outcome assessor is blinded. The participant and the investigator are not blinded. Blinding may not be possible always. So in sometimes it may be challenging to do blinding. For example, you have a situation where one group receives drug therapy, the other group receives a kind of a surgical option in addition. Then it becomes difficult to blind. Then you might have to go for a open label study. So any study where where randomization is done, but blinding is not done, you call it as an open label study. Okay. Now, even though you take all the effort to do blinding, sometimes, in spite of all your efforts, blinding might be unmasked. Okay, so how does that get unmasked? Especially, it happens when you're doing trials where one group receives a medication that has very distinct side effects, and the control is placebo, where those side effects are very unusual to happen, unusual to occur. So then, even in spite of blinding, at one point, the patient as well as the investigator knows which group of participant can be belonging to. So there are some trials where usually what they do, in order when they report the trial in a journal, they go one step ahead and say, at the end of the study, we tested the effectiveness of our blinding by asking the patients what they felt as which arm they belong to. And they'll see how much correlation is there between what is the reality and what the patient felt. So if there is not much correlation, that means the blinding is good. But if all the people are able to accurately predict whether they were receiving study drug or the control drug, then it means the blinding was not very effective. Now, another question that we ask in this context is placebo. So obviously, when you're having a blinding, and it's especially done in placebo-controlled trials. So, what exactly are these placebos? As you might know, placebos are inert molecules which do not have any pharmacological action, and so they are usually used as a comparator, as a control. Uh, One group receives the test drug, the new drug, and the other group receives the control drug, which is the placebo. Okay. Now, placebos, there are a lot of questions, a lot of ethical issues concerning placebo. So some people go to the extreme extent of saying placebos should not be used in clinical trial. Okay, but there is a lot of scientific basis on the utilization of placebo. So one is placebos. the reason placebos are used is it is almost one of the best way by which you can really say, scientifically speaking, that your drug is better than a situation where a patient does not take that drug. Okay, but ethical challenges are there. Especially, you cannot give placebo in situations where you have a disease where it's very fatal or very severe disease, and where one group receives only the study drug and the other group receives only placebo. That becomes highly unethical, because when already there is a best available standard of care therapy, at that point of time you deprive one group of people with that standard of care and give them only placebo. It becomes highly unethical. It's almost never done in today's world of clinical trials. So then, in which situations do you use placebo? They are widely used in two situations. One is as an add-on study design, where both the groups receive the standard therapy. In addition, one group receives test drug and the other group receives placebo. So this is called as placebo add-on study designs. Okay, add-on placebo study designs. This is very commonly used, particularly in cardiovascular clinical trials. It's very common to see this practice being done. And the other situation is when you have a disease that is not very severe, a mild kind of disease. Let's say migraine that is of mild nature. Okay, in this situation, you could go ahead and do placebos, because there is a situation where you can, in case the patient has a very severe headache, you can always have a escape rescue medication that is given to such patients. So in such mild illnesses, you can give placebo. And also another situation when you do not have any treatment, like for example, right now we have vaccine trials that are going all over the place. So many of these vaccines, like for example even COVID-19, we do not have a COVID-19 effective vaccine in the market. So since we do not have a COVID-19 effective vaccine in the market, okay, it makes sense to have placebo as a comparator so that we know whether the COVID vaccine is working or not. Another important aspect when you're planning or doing an RCT is the choice of endpoints. So what exactly is endpoint? Endpoint is a measure which is expected to be modified by the study intervention over a time period. So what is the measure, the variable that you're using to modify and to say that your study drug works? For example, in a cardiovascular trial, I might say by taking this drug, the mortality will reduce at the end of 1 year compared to those who do not take the drug. Okay, or you could have a situation where you're saying this drug will change or reduce the blood cholesterol at the end of 6 months by this much percentage, or reduce the HbA1c by this much percentage. So that is what is called as endpoint. Okay, a measure that is going to be modified by the study intervention over a particular time period. Very often, outcomes and endpoints are confused. So outcome refers to the measured variable, whereas endpoint refers to the complete analyzed parameter. An example I'll give you. For example, in a diabetic trial, HbA1c is an outcome, okay? But the actual endpoint is a change from baseline to 12 weeks in the mean HbA1c. So this is the real difference between an outcome and an endpoint. But practically in many textbooks or even many research articles, you will find that both are interchangeably used. But there is a difference between the two. Some people do not differentiate these two and they freely interchange these two. So do not get confused there. So I think I'll skip this for the sake of time. So you can have different types of endpoints. You could have a endpoint that is clinically meaningful, that directly impacts the patient, how the patient feels, functions, or survives. Or you could have a endpoint that is a surrogate endpoint. A surrogate endpoint is usually a laboratory measure or a physical sign that is acting as a substitute to the original clinically meaningful endpoint. Now you may ask, why are we doing surrogate endpoints? The reason we are doing surrogate endpoints is surrogate endpoints are convenient. It's easier to do a lab measurement, because in a lab measurement you can get that result within say a few weeks. Okay, whereas to see the clinical improvement, that survival is there, you have to follow the patient for 5 years. So if you want quicker results, then and you have a study budget that is lesser, and you want to have a smaller sample size, then very often we go for surrogate endpoints. Okay, examples of surrogate endpoints here, you have hypertension. So for hypertension, arterial blood pressure is actually a surrogate endpoint, because what we are concerned about mainly, clinically speaking, is not the blood pressure per se, but the risk of cardiovascular events, the risk of cerebrovascular accidents, the risk of MI, the risk of heart failure. Okay, and similarly for HIV, it is the CD4 count which is the surrogate endpoint, or the viral load. Okay, whereas the real our hard outcomes will be the actual complications of HIV. So how to handle this? Another common issue that happens in clinical trials is the dropouts. Okay, the dropouts. So what exactly are the dropouts? What exactly are dropouts? The dropouts are basically people who get randomized into the study, and at some point of time they withdraw, or they are not willing to participate, or they have side effects. They kind of come and go, and then they at one point they fall off and they do not be part of the study. Or there are some people who will just get randomized and not come back even for one visit. Okay, so how do you handle all these kind of situations? So you have to carefully decide how to handle this, because you cannot just say, okay, I'm doing a clinical trial, I will only take people who complete my study. All those who dropped out, I will leave them out. Okay, because it is possible that when you leave them out, you will remove a lot of people who are actually dropped out because of adverse events. Okay, so all this should be investigated thoroughly. And then I'm closing now. So we have some other designs called as crossover clinical trials and factorial study designs. So briefly, I'll just tell what they are. So factorial design is essentially a trial where you are testing the effect of two or more treatments simultaneously using a combination of the treatments. Okay, for example, I'll just show it with an example. You have growth hormone and pioglitazone are being compared uh, are being given to patients with viscerally obese adults with impaired glucose tolerance. Okay, so this is called as a factorial clinical trial. So what exactly they have done is, I just want you to focus on this last row. So there are four groups. One group receives placebo pioglitazone, growth hormone placebo. Another group receives pioglitazone plus growth hormone placebo. The third group receives pioglitazone placebo with growth hormone. And the fourth group receives both growth hormone and placebo. So when you, in the same group of people, you're looking at two different interventions, and the interaction between the two drugs also are being compared. Okay, so that is a factorial clinical trial. And then you have the crossover study design where you randomize people to two groups, but at some point, what you do is you stop them of the drug, give a washout period of a few weeks, and then those who received treatment A in the first period, in period two, they are switched over to treatment B, and those who received treatment B in period two, they are receiving treatment A. And so the difference between a patient's response in treatment A is compared with the same response in treatment B. So each patient acts as their own control. So this is called as a crossover study design, especially useful in chronic diseases that do not have much of relapses and remissions or exacerbations. Finally, the quality control is a very important aspect that has to be kept in mind when you're doing clinical trials. Maintain an SOP, follow them, train the investigators, ensure that there is not too much of subjectivity involved with respect to outcome measurement with the way the data is handled. So, as you have heard yesterday and today, randomized control trials are the apex of evidence-based medicine, particularly randomized trials that are well designed, well conducted, and well executed. So these randomized control trials, they are much superior than the observational study designs because of the obvious bias and all that is there in the observational studies. And most of the drugs that are today approved in the market are there because well-designed randomized controlled trials have proven that these drugs are working in those conditions. Okay, so they form the gold standard of evidence-based medicine, and of course, systematic reviews of these RCTs form the best form of evidence. So I think I'll skip this. So in conclusion, the challenges of RCT, I just want to say this and close. It takes a lot, in spite of all the good things that we have been talking about RCTs. Why is it that still RCTs have a problem? Is that it takes time for doing RCTs. In fact, if you go to clinicaltrials.gov, you will find that there are a lot of trials which have started and could not be completed. Okay, for various reasons. Okay, it could be they could not recruit enough subjects because the disease is rare, or there are many other practical challenges in doing trials. So because of which they could not complete it. Okay, it's costly. It takes, in fact, to bring out one phase 3 trial, it could almost cost around $750 million. May not be possible to complete it sometimes because of the very rigid inclusion exclusion criteria. At the end of the day, you're not even able to find patients who are exactly fitting that criteria. Protocols are extremely complex, particularly if you look at oncology, cardiology. There are very complex protocols that becomes very difficult of people to participate in such long duration of time. And a lot of regulatory challenges are also there. Uh, Regulatory environment is more stringent. In fact, I once read that apart from the airline industry, the second most regulated industry is the clinical trial industry, because we are dealing with lives here. So data is very stringently regulated. And RCTs are also reflecting a artificial environment. They are not reflecting a real world practice. So because of that, it is sometimes uh, people criticize randomized controlled trials for that reason, that it is not really telling the truth, because this is a very ideal hypothetical environment you're doing a trial, whereas in the real world the drug may not be that effective that as was seen in the clinical trial. So that is why these days people talk a lot about real world evidence, so that to really see whether in the normal clinical hospital setting, do these drugs work as effectively as what we saw in the uh, RCTs that were done by the pharmaceutical industry. And it is also important that we use patient related outcomes rather than other outcomes which are more of surrogate in nature, which may not have direct clinical value. So these are some of the challenges with RCTs, but nevertheless, whatever said and done, whatever evidence, I mean, whatever treatments you see in the market today, especially when it comes to interventions, they are all there because of well designed, executed RCTs. And one of the best things that you can do to foster the growth of medicine is to do RCTs and really prove whether the drugs that you're using, they are effective or not, whether they are useful or not, whether they are safe or not, by using a RCT design. So with this, I close. So if you have any questions, you can put it in the chat box. We'll try to take it in the Q&A session. Okay. In the interest of time, I'll just take one question and then we'll go to the Q&A. Okay. The difference between allocation, concealment, and blinding. So as I said, blinding refers to the process of making the drugs or the interventions look similar between the two groups. Whereas allocation, so blinding refers to drugs, okay, whether the drugs are similar or not. Okay, so practically speaking, how do you do blinding? You make the placebo look similar to the study drug. That is what is blinding. Okay, in its external appearance as well as its packaging. Whereas allocation concealment is a process of concealing the identity of the, a process of concealing this randomization sequence from the investigator who is going to recruit subjects. It has nothing to do with the study drug. Okay, It is to do with the sequence, not the medicines that you're having in hand. Understood? So this is the essential difference between the two. There are lots of other questions. I'll try to answer all these questions in the Q&A. In the interest of time, we'll stop this here. We'll go to the next session. I request Kavya, you can just introduce the next speaker, Dr. Satyajit.
Thank you so much, sir, for such a colorful and stupendous session. So our next session is on the topic protocol writing, which has been taken by Dr. Satyajit Mahapatra. So to talk about, to introduce about the speaker, so Dr. Satyajit Mahapatra is currently working as a professor, Department of Pharmacology in SRM Medical College, SRM University, since May 2019. He passed his undergrad MBBS in 2002 and master's in pharmacology in 2010 from Government MCH Medical College, Berhampur, Odisha. He is the internal faculty of AFIH, the Associate Fellow in Industrial Health course in School of Public Health, SRM University. He is pursuing his PhD in pharmacology in SRM University and is also undergoing advanced course in medical education course recognized by MCI. He has 16 years of rich clinical experience and 10 years of teaching experience. He has a rich experience in conducting research, clinical trials, project related to pharmacology and pharmacotherapy. Insightful understanding of good clinical practice guidelines, Schedule Y, the Drug and Cosmetic Acts 1940, the Drug and Cosmetic Rules 1945, and patent laws. He has successfully conducted many clinical trials of rotavirus vaccine, pentavalent and quadrivalent vaccine by multinational pharmaceutical companies as a principal investigator. He is also famous for Covaxin trial for phase 1 and 2 and 3, which has been conducted in SRM Medical College as a principal investigator. And he is a life member of Indian Science Congress and Indian Pharmacological Society. It is an honor to have you here, sir. Please take over the session. Thank you.
Hello. Kabir, can you hear me?
Good morning, sir. I can hear you.
Good morning. Can you see the slides?
Yes, sir. You can please put it in the full screen. Thank you, sir.
Okay. Thank you. Thank you, Kabir, for the nice introduction. Good morning, one and all. At the outset, I'd like to thank the organizing committee, especially Dr. Melvin and HOD of Pharmacology, Dr. Jamuna Rani, for giving me this opportunity. I think the participants are in a flow for last one day yesterday as well as today's morning session, and it's quite a nice session by Dr. Melvin regarding the study designs. Next, we'll discuss about the protocol writing and its key elements. All of you know that writing a research protocol is one of the most challenging part of research. And the purpose of this session is to summarize the most important steps and necessary guidelines for producing a standard research protocol. And suppose you plan to submit your project or a proposal to acquire a grant in the field of research, the success depends on how good quality protocol that you are writing, and that is important aspects of this protocol writing. And so the quality of protocol is required to get to achieve a success in grants and other things, academic achievements. Clinical research is conducted according to a plan. So if you plan properly, then it demonstrates the guidelines for conducting the research, then the outcome will be better. So it illustrates that what will be made in the study and how it will be carried out. So if you plan your protocol properly in the beginning, you will get a good outcome. And that means the outcome is a standard protocol, right? So if you see in Lewis Carroll's Alice in Wonderland, the king says to the white rabbit, "Where shall I begin, please your majesty?" And the king says that you begin at the beginning, then go until you reach the end, then stop. So here he could have been telling an investigator that the process of crafting a clinical trial protocol. So you start with a clear idea what you are going to do. So until you reach your destination. So protocol is an very important aspects of research. So what is research? Research is the systematic collection, analysis, interpretation of data to answer a certain question or solve a problem. So if you want, you have a question, you have a research question, you have a problem, and then you plan out how to conduct a research for that particular question, and then you plan your methodology, then you plan your collection of data, how you're going to analyze your data, and how you're going to interpret the data. So if you compile all these things in a proper format, it becomes a protocol. So before you go to writing a protocol, you should know what is the life cycle of a research, right? So life cycle of research is, it takes you through various steps in which you would do a research study. So please keep this in mind, that these following steps of life cycle is vital to have a good study which is logical, which should be focused, and we should be efficient in a way that you are going to conduct a research. So whenever you're thinking of writing a protocol, or if you're planning to design a study, or make sure that please make sure that you do not jump out of these steps of the life cycle. So you have to follow one by one. So first one is how you identify the needs of the problem. That is very important. So you identify the problem, and based on the problem you formulate your research question. Then, on based on the research question, you formulate your research study objectives. That is very important. Then you plan your methodology, which is the major part of the research. So, based on the methodology, how you are going to collect and analyze data, then you interpret your data and draw a conclusion, and you can formulate the recommendation, and then inform the stakeholders. Again, based on this lacunae or any pitfalls there, again you identify the needs. Again, one research question can be developed. So it is a complete life cycle of research. It goes on and on and you should follow the steps one by one. Do not jump from research question to conclusion, or from this to that. So it should be step by step. That is very important. So till now, you have gone through all the aspects of how to design a study, research study, starting from thinking of a research question, doing sampling, then selecting the research participants, then doing measurements for exposure as well as outcomes, and if you look for any human subject protection like ethics issues. So when all these structures come together in a research in a written format, that is known as a protocol, which is basically a written plan of a study. Now, why do you want to write a protocol? So there are many aspects of a protocol. So you need to clarify the research question. So you have formulated a research question, whether you can justify that yes, the research question is valid, and you just want to clarify that. So you have to write a protocol. You have to compile the existing knowledge. So studies should be done. Whatever studies has already been done by other investigator, you want to compile. Kavya, is it okay? Can you hear me?
Yes, sir, I can hear you. Please
Okay.
proceed.
Yeah. To compile the existing knowledge, and whatever studies has been done by other researchers, then to formulate a hypothesis and objectives, to prove the hypothesis whether it is true or false, and to decide about a study's design, then to clarify the ethical consideration, and if you want to apply for any funding, you have to write a protocol, and to have a guideline and tool for the research team. So all these aspects are the purposes of drafting a protocol. Now, who reads the protocol? That is very important. Suppose you are writing a protocol, you should know that the protocol language and content should be very clear, and because it will go to various agencies, like it will be read by other physicians of scientific committee, other members of the ethical committee, scientific reviewers, your peers or colleagues, or if you are applying for grant, the grant agency will read your protocol, and if you are attaching an informed consent form in the protocol, the language should be understood by a layperson. So considering all these points in your mind, you should write a protocol very clearly, very neatly, and so that it will be read by all the stakeholders. Now, coming to writing a research protocol. So there are various components of a research protocol, like starting from title and abstract, then introduction, then M's objective, uh, and then content, the context of, you have to write the review of relevant literature. And then the major part is the methodology, your research question, your study design, and then the description of methodology, and what is your plan of analysis. And that in the end, you can write your references, like you can put the citation, the citing style, the citing journals, whether it is from any books or internet reference, all these things you have to consider properly. Now, coming to the project title should be accurate, short, and concise. So once someone reads your title of your project, he should able to understand. Okay, at one glance, it should be clear that what is the study about, who are the target population, and where is the setting of the study, where it is done, how it is done, whether it is hospital based or community based, and when it is launched, the time also. So important is, if it is descriptive, you should make the main objective clear, and you should mention the target population. And very important is keywords. So keywords for referencing, it should be there in the title of the project. So why do research title matters? Because as I told you, articles with interesting descriptive research titles draw in attention. So if you search in the search engine, you go to PubMed and search for something interesting, you put some keywords. So lots of articles will be displayed. Now you have to find out which article is interesting. So when someone searches and find out an interesting article, the title, it will be referred many times. So you have to be very clear about your title, which draws attention. Second is, a good title predicts the content of the research paper. By seeing it, the researcher should know, the others should know that, okay, this is the content, this might be the content of the research paper. Third one is, a good title should be interesting to the reader. Okay. Fourth one is, it should reflect the tone of the writing. So by seeing that title itself, they will know, okay, this could be the logical flow, the logical tone of the writing in the article. Then it should contain important keywords that will make easier to be located during a keyword search. So I'll give you an example. Suppose you want to see whether meditation makes nurses a better communicator. Okay. So here what I'll do is, I found three keywords. The first one is nurses, meditation, and communicator. So with these three keywords, I have put in the search engine, and I got these four results. You can see the first one is benefits of meditation for nursing profession, a quantitative investigation. The second one is why mindful nurses make the best communicator. The third title is meditation gurus, and the fourth title is nurses on the move, a quantitative report on how meditation can improve nurse performance. So out of these four, I think the fourth one is showing a good, it's interesting and showing the all the aspects of a research title. Like it includes the topic, it includes the method, that is the observational study, the sample, as well as the result of the study. So meditation makes nurses perform better. So this is the conclusion. There is result of the study is that meditation makes nurses perform better. So this is the result of your study. A qualitative study of mindfulness. So qualitative is the method what you have used among nursing student in the sample or the target population. Now, before writing a full protocol, you have to write a project summary of your protocol. That is, you have to write a concise one-page, 300 words project summary, which stands on its own. So someone reads that, he will understand, okay, this will be the content of the protocol. So if already made a concept paper, write more details about it and summarize the central elements, like what is the rationale for the study, what methodology you are going to use, what is the population, and what is the time frame, and what is your expected outcome. Now, coming to the statement of problem in the introduction part. Now, when you're writing a statement of the problem, why the research is needed and what is the relevance of the result that you are going to find. Now, logical flow of statement should be there. So whenever you're writing the problem in the introduction part of the background, the statement of problem, it should be logical. That should be logical flow of the statement. First one, the magnitude, frequency, and distribution of the disease uh, or the research problem. So affected the where it is more relevant or more prevalent, the population groups affected, the ethnic and gender consideration, then what could be the probable cause of the problem? So what is the current knowledge of the problem and what is its causes? Is there any consensus among the literature or the articles you see that, or is there any controversy, or is there any conclusive evidence? So this you have to find out. Then what could be the possible solution? So in what ways have solution to the problem has been attempted, and what can be proposed, and what are the results? So this is your possible solutions. And then the unanswered question, what remains to be unanswered, what remains to be answered, what areas have not been possible to understand, determine, verify, or test. So based on this unanswered question, you can go with your research question. Now you have to do a literature survey, right, for the statement of problem. So while taking this part of the protocol, when you are going to survey your literature, you should know that it should prevent duplication of work which has already been done. So why you need to do a research literature survey? You should know that whatever plan you have for your research, it should not have been done by some other researcher. So it will prevent the duplication. Second is it clarifies what others have found addressing the problem. Suppose you are planning a research on diabetes or hypertension on some new drug, whether the same thing has been done somewhere and what result they have got, whether it can be addressed further. It familiarizes with potential methodology. So once you go to literature, you can see, okay, what concept or what methods they have used for their research, what are the errors they found. So based on that, what are the limitations? So all these things, based on that, you can have a clear idea. And it should convince that research is needed. So that is the importance of literature review. I think yesterday, Dr. Subramanian took literature review. You have to see the citation, because in literature review part, in the when you write your background and introduction, you put the citation of your references. So you have to double check all the citation, because most of the time the common mistakes occurs there, and misspellings will be there, wrong initials, wrong journal name, and years of publication, and wrong volume numbers. So to do that very carefully. Now, justification of research, it should be convincing statement for the need to do this research. So how does this research relate to the priorities of the region? The same thing as I told you in the last slides. So what knowledge and information will be obtained if you do this research? What is the ultimate purpose that the knowledge obtained from the study will serve? How will the result be disseminated? And how will the result be used? And who will be the beneficiary? Now, you have to keep in mind when you formulate a research hypothesis, what is the independent variable and dependent variable, and what type of design you are going to do, whether it is an intervention or observation. So you have to describe the relationship between the independent variables as well as dependent variable, that is your outcome. And you determine the data to be collected and the type of analysis to be conducted. Now coming to the research objectives, it should be very simple, it should be specific, and it should be stated in advance. So when you formulate your objectives, you have to you have to focus on your study and narrowing it down to the essentials, and you have to avoid collection of data which are not strictly necessary for understanding or solving the problem. That you can have secondary objectives, but the primary objectives should be, it should be focused on your study and whatever data which is conclusive for your research study. So you have to organize the study in a clearly defined part. So you can put a general objective, and then from the general objective, you can derive some specific objectives. As I told you, objectives should be logical, coherent, it should be feasible. You have to see whether it can be done, it should be realistic, and it should be defined in operational terms that can be measured, measurable, and relevant. So it should have SMART components, right? Specific, measurable, achievable, realistic, and time bound. And you can use some action verbs like to determine, to compare, to verify, to calculate, to reduce and to describe in writing a research objectives. Now, coming to the important part of your protocol is the methodology. So, as I said, we need much more details, and major part of your protocol is actually the method section. So, where you give the whole detailed plan of how you are going to conduct the study, starting from the study design, describing the intervention if you are doing an experimental study, or describing the study population, writing out your operational definition. So, very important. It assures that hypothesis will be proved or disproved using the right tools, and it presents in a detailed strategy how the objectives are achieved. So these are the outlines of your methods. You have to describe in details about your design, the intervention, if you're planning an intervention study, and what are the study population, and what are the operational definition, what are your sample methods that you are going to use, and how did you calculate the sample size, that also has to be clearly defined. And then what would be the data collection method, and then what would be the tools to do that, and what is your analysis plan. And then a detailed project implementation plan you can write, like how you're planning to maintain the quality and finalizing the human subject protection and ethical issues. So that's all the final aspects. Now we'll discuss one by one of the steps in more detail, how you can develop a protocol. Now coming to design, the decision of study design needs to be based on the ethics, logistic consideration, economic aspects, and scientific thoroughness. It explains how the objective lead to the indicators and to the study design. Suppose, as I told you, if you are in the study design, if you are planning to have an experimental study or interventional study or observational study. So are you planning to do a RCT, or you are planning to do a cohort study, cross-sectional study, or case control study, or ecological study. Now, intervention, suppose you plan an intervention, how it is planned. Are you going to plan a clinical trial, or you are going to plan a community intervention? So it describes the treatment applied to the intervention group as well as in control group. So that is very important. Then description of the intervention. Now, if you are going to do an experimental study, whether it is a clinical trial or community-based intervention, so accordingly, you will describe your interventions. Who is going to be intervened? That is important. What population, which population, what age of the population is going to be intervened, and where it will take place. What exactly your intervention? You have to write in more details. What would be the time period over which the intervention is going to be applied? So that also is to mention. And the whole procedure of whether the clinical trial or community intervention has to be described in details. So in this section, you have to write who is responsible for the intervention, whether it is a study nurse or is the investigator who is going to intervene, and what intervention you are going to give, whether it's a drug, it is a vaccine, or it's a information, or it's a surgery, whatever it is, and where it will take place, the exact location where the intervention will take place, and what are the activities to be performed, and what frequency and intensity you have to perform. Inclusion and exclusion criteria you are going to be using to select the population. That is important. Either you may be, it can be a hospital based, or it is a community based, or it is a population based according to your study design. Now, up to over what time of period you are going to select these people. That's also you have to mention whether you are going to enroll these subjects for 3 months, for 6 months, or 1 year. So the time period is to be mentioned. The geographical location from where you are going to select these people. That also, if in and around SRM Medical College, Cotton Klatur district, Cotton Klatur town, or you're going to do the whole Chengalpet district. So all these things you have to mention in it mentioned clearly. Then individual characteristic of the people that you are going to select into your study. So study population is the general population. So you are going to do a study in the general population. So that is the study population, whoever is according to your target area, then target population. Then a part of them you are going to sample based on your sample size, based on your inclusion exclusion criteria. That becomes your study sample. Okay. So study population is the target population. So study sample is out of that, based on your inclusion exclusion criteria, you have to select the study sample and based on your number. So when you the points to be considered to write a good eligibility criteria. When you write inclusion exclusion criteria, it is very important. You have to note down this the criteria, the points to be remembered. The number of eligibility criteria should be kept to a minimum. You should not put like 50 criteria for inclusion or 50 criteria for exclusion. It should be minimum. It should be absolute necessary to ensure the scientific validity and patient safety. So it should include those criteria which are absolutely necessary. Eligibility criteria should be clearly defined and verifiable by an external auditor. So it should be clearly defined. It should be straightforward and unambiguous. I'll give an example here. Suppose you want to exclude a pregnant woman from the study, from your study. So how are you going to write an exclusion criteria? The first criteria I am going to write is pregnant and or nursing women are not eligible. All women of childbearing age are required to have a negative pregnancy test. This is the second criteria which I have written, the second alternative. The third one is a female participant of childbearing age and planning to avoid pregnancy, bracket, use of an effective contraception or abstinence from the time of study enrollment till 4 weeks after the study period. So this gives a clear picture of an exclusion criteria that a female of childbearing age and who is not planning to pregnant from the time of enrollment till 4 weeks after the study period. So this is clearly defined. So this is the correct exclusion criteria for a pregnant lady or a nursing mother. Now coming to subjects and participants, depending on the type of study, answer the following question. What are the criteria for inclusion, exclusion, or the selection criteria? What are the criteria for exclusion? And in intervention study, how your subjects are going to be randomized and what are the randomized procedure? Now Melvin told what are the different randomization procedure and what are the criteria for discontinuation or follow-up, loss to follow-up or dropouts. So those criteria you have to clearly mention in the protocol. Now you have to write your variables also. Now this various operational definition we are going to be used in your study should be clearly defined. You have to spell out how you are going to define and measure your key outcome measures and what would be your key exposure measures, what would be your operational definition for this, and you should be very clear and specific in terms of this definition. So you can see if you suppose one variable is age, the operational definition is age at last birthday. So it should be clearly defined. The definition is age at last birthday. And scale of measurements is it should be in months. So you can write like how many months, whether it is 12, 180 months or 200 months or whatever. There you have to clearly put it in the measurement scale. Similarly, hemoglobin concentration in capillary blood measured by hemoglobinumeter. So this is the definition of the variable that you have used for your study, that is hemoglobin percentage, and it should be in grams per 100 ml. So percentage, it's rounded to the nearest gram. So these things you have to clearly define in your protocol. Now coming to sampling procedure, what type of sampling that you are going to be using, whether it is random sampling or it is a non-randomized sampling like simple, systemic or cluster sampling. I am not going to details because already it is taken, and how you are going to select the sample in actual practice. And you have to provide reference if you have taken any. And if you are doing if you are doing a RCT, randomized control trial, then you would be using randomization. So you should explain what kind of randomization technique that you have used and how the study subjects are going to be randomized into intervention group and into the control group. Sample size calculation is recommended for economical and ethical agents. So you have to write all the parameters which are used to estimate the sample size. What is based on the study objective, the methodology, and what software, like how did you calculate your sample size, whether you have calculated using a software or using a formula. So that has to be mentioned, and you have to give references if you have taken this thing. Okay. Now coming to data collection and data analysis. Now what are the data, what are the parameters you are going to take for your research. So list the data that will be collected. What are the socioeconomic data? or if you are going to individual data or your signs and symptoms. So these things should be clearly defined and it should be listed out. And how we are going to collect the data, who is going to collect the data, whether it is a PI is going to collect, it is a co-investigator going to collect, the staff nurse is going to collect, the other clinical research coordinator is going to collect. So that is to be mentioned. And how you are going to collect, what type of instrument to be used, whether you are using any abstraction form, you are doing an interview, this is a qualitative research. Okay, so if you're doing an interview and taking out the data, face-to-face interview, focus group discussion, or questionnaire based, so what type of type of instrument you are going to use for the study, you have to mention. Coming to measurement of observation, describe how, when, and where the observations are made, and the this I told the instrument used, like questionnaire, the interview, the lab test, or the clinical examination. So if you are doing a clinical examination, what gadget or procedure you used to be clearly mentioned. Then the data analysis. So you have collected the data, you have to enter the data in the either in a computer or you are keeping it manual. So the mode of data entry is important, whether you are keeping it a manual data or if you are entering in a Excel in a computer based. Uh, what are the software used? So to analyze the data, that also is to be mentioned. And for descriptive stage, suppose prevalence and incidence, you have to use this. And for analytical stage, univariate or multivariate analysis, what type of analysis you are going to use. Do not mention that suitable analysis will be done for the data collector. You have to clearly mention what type of variable, what statistical analysis, statistical method you are going to use, and for what software is used. Clearly it should be mentioned. Don't mention suitable analysis will be done. That is completely a wrong statement. Now data management and analysis. So based on the objectives, you have to code your variables and the types of variables. And what is your analysis plan depending on the type of variables. What are the statistical tests you are implicated, and style of presentation, whether you are putting it in tables or graphs, and how you are going to monitor, keep the data safe, and how you are going to store the data. So quality control is important for the data. So before you enter the data, you have to double check whether your data is correct or not. Consistency in checking. You should have a data monitor and verification procedure for the source data. Now, one more important aspect in this protocol writing is the ethical consideration. If you are involving human subjects, ethical consideration should be taken care, because it is important aspects of the biomedical research. And your local ethics committee will ask you question. They will verify your protocol. They will see whether you are. They'll look into the important aspects like whether how you are going to safeguard the data or the confidentiality of the data that you have generated. Are you going to, is there any possible harm, side effects, or consequences of your intervention? They're going to look into this. Is there a right of discontinuation at any time has been addressed? That is voluntariness. The subject can withdraw at any time during the participation. Role of data safety and monitoring board in the trials. Clinical trial we put as DSMB, the role of DSMB. And then alternative treatments and approaches, and in the trials, and what are the incentives and rewards you are going to give. So this important document you are going to attach, right? Informed consent form, and you have to outline how, when, and where the subject will be consented. So informed consent form, it should contain the justification of research, why you are going to do this research, who is responsible for this research, the principal investigator, outline of your study. So you put your methods, the risk involved in the study, the benefits involved in the study, whether it is insured, and how you are going to keep the confidentiality of the study, and the voluntary participation is important. So a separate consent form is required, and when you're preparing a consent form, it should be very simple, and it should be in the mother tongue of the or the language which the participant can understand. Okay. So ethics checklist is you should answer all the potential questions regarding the ethics, as I told you, and it should focus on the pros and cons of the research design, the selection of subject, the measurement and outcome assessment, and it should discuss the advantage, disadvantage of the subject or communities involved. It should discuss physical, social, and psychological implication of the research, and it should discuss the confidentiality. Then comes the reference and bibliography. References, you can use either Harvard style or Vancouver style, and you can make use of the software like EndNote and Reference Manager, and you can put according to how the number is, the reference is assigned. It's cited either in the bracket or round bracket, or you can put it as a superscript. Now, the last one is the project implementation plan. So this section, it details the steps that you are going to use to implement the study to ensure the data quality at all stages of the study. Now, how you are going to collect the data, the way you are going to enter the data and analyze the data, and how you are going to report this study results. So this is the place where the roles and responsibilities of the investigators uh, has to be mentioned clearly. Okay, so who would be responsible for this project, like in terms of administrative responsibility? Suppose it's a clinical trial, the head of the institution is administrative responsibility if it is, and then who are involved in the technical, the principal investigator, the other co-investigator, the coordinators, the study team, whoever is involved, what is their role and responsibility is to be clearly defined. Then all logistic to be used during the data collection has to be mentioned here, right? And then the timeline for your study, where how much time you are going to take, whether it is a 6-month study or 1-year study, and different phase of the study, starting from designing the instruments and getting approval from the ethics committee, and then doing a pilot testing, then the actual collection of stuff. So you need to give a timeline over a period of the study. It is very important. Who is going to do, when is going to do, where you are going to do. So for each one, suppose you have to plan like Jan, Feb, March, April, May. So it's a 6 month. You have to, if clearly, if you put a project timeline, I think that will be very helpful for those who are planning to do any research. If you're writing a protocol, please put your project timeline, and then who are the staff who are going to meet, like who is going to design the questionnaire, and who are going to go to the field workers, and then pilot questionnaire, the data, who is going to take the data, and what is the timeline for data collection, then who is going to analyze the data. So all these things you can clearly mention in the project timeline. Suppose you are planning a study for grant, either it's the institution or uh, the extramural fund. So you have to break down your items. So based on your personal use, how many persons are there in the study team? What are your consumables? What are the equipments, supplies, communication, funds for the patient, data processing? All these things you have to break, and you have to give a justification of that. So you have to justify each item, what you have put in the budget section, and what is your work plan for the study. And these are the annexes you have to attach in your protocol. The SOPs, if the SOPs are there, what is the training framework for the field workers, if the participant recruitment material, the consent form, if some adverse event happen, how we are going to manage that adverse events, and study management form. So these are the, as I told you, case report forms, questionnaire, then PIS and CV of the investigator is to be attached. So these are the basic outline of a research protocol, the title of the research project, then project summary, and then statement of the problem, like justification, then justification of and use of your results, the objectives, then theoretical framework, then research objectives, general and specific, then your methods, then plan of analysis, then references, timetable, budget, annexes. So this is the outline of a research protocol. So once you put all these items together, you need to form it up. You tie it everything together. Now this is not a one-step process. It goes through several drafts. Please remember, once you write a protocol, it is not the final protocol. It has to go through several drafts. Draft number 1, 2. That's why the version changes when in clinical trials also, because it is reviewed by many committees. It goes to the expert committees, the subject experts committee in the regulatory, and then they give advice, and then based on that some modification occurs. It goes to the ethics committee. Ethics committee gives some suggestions, then modification occurs. So it goes through several drafts, and it is actually a good idea to be reviewed through your through your peers. So once you write a protocol, you give it to your peers or colleagues or the subject matter experts, and then get feedback from them, and then you can revise your protocol. So one more time, if you submit to ethics committee, they themselves have some suggestions, because they want something to be changed or revised in your protocol, so that has to be incorporated. So that becomes one more draft, right? So you will be doing that work again and again, and which will lead to another draft protocol. So by the time you are ready with your final draft, you will have multiple drafts with you, right? So it is always a good idea to keep everything, to archive all these drafts, so that we will know how your study is evolved and what are the different ways that you have planned for your study. So starting from the draft one to the final draft, you keep everything. You can know that what suggestion was given by your peers, the scientific experts committee, or what suggestions was given by the ethics committee. So you archive and then you prepare your final protocol. So these are the 7 steps of a successful protocol. So you have to identify a question, objectives, then concept paper, then prepare a table, draft protocol, then prepare your instrument and annexure, then submit to peers review, and then ethics committee approval. So before you submit, you do a proofreading of your protocol. That is very important, because working too long for on a protocol may habituate eyes and brains to do mistakes. So because they have been there for all along, and they are seeing those objective, their problems, the methods. So you can go with a spell checkers, and a document should be checked with a software, or it can be proofread. So according to a research at Cambridge University, it doesn't matter in what order the letters in a order. The only important thing is that the first and last letter be at right place. Here you can see, even if the letters are different, disorganized in the words, still we are able to read. Okay, so while writing a protocol, we miss all these things. The spelling errors are there, so you should do a spell checker before you submit to ethics committee. So how do you know that that's a good protocol? Whatever you've written, it's a good protocol. Is it adequate to answer the research question or to achieve the study objective? Is it feasible to the particular setup for the study? Very important, whether it is feasible, we can do it here. Does it provide enough detail that can allow another investigator to do the study and arrive at a comparable conclusion? So if you are doing a study, if another investigator plans to do the study, will he get the same comparable conclusion as you have got? So based on this, you can judge that this is a good protocol. Now, what we do in the common mistakes in writing a protocol is we incorporate insufficient elements regarding the study. That is very important. We never do a good literature survey, so we do a very, like, we give insufficient elements. We give insufficient explanation for implication of the problem. We suggest more work than what can be practically done during the study period. It's not feasible, but we project like we can do this, we can do that. And then unpowered sample size. More sample size is more or sample size is less. So we have to give a good power for the study, and invalid and unreliable instrumentation should be tested. So to conclude, a proposal is considered successful when it is clear, free of typographical errors, accurate and easy to read. You can understand the steps in developing a research protocol. Extra time should be spent to write a good protocol, because it will save failures at later stage. So you spend your time in doing a literature review, spend your time in designing the study, spend your time in how you're going to take your data, collect your data, how you're going to analyze the data. So take time before you submit a protocol. Because if you submit it's not a good written protocol to a ethics committee or scientific committee, they will reject you again and again. It will be rejected, and it will, like, you will have you will not have a good mental states, like, to submit it or to write it again and again. So you spend time in writing a good protocol at the beginning, so that you will not have failure. And if you it's poorly prepared and not adhered to the guidelines, that's chance of selling your idea to the reviewer of granting agency also. If you're planning to get some grant, it will be rejected at the first place if you are submitting a poorly prepared protocol. So protocol development is a multidisciplinary process. Please remember, you can involve if you are planning to do it with other department. Also, you take help of the other department peers, colleagues, and then you plan your protocol. Unfortunately, there is no control experiment for writing a protocol. So how you don't have any control to judge that this is a good protocol and our protocol is as superior or as non-inferior to the control protocol. Thank you.
Thank you, Dr. Satyajit, for this elaborate discussion on protocol development. So I think as beginners, and even if you consider yourself as a seasoned investigator, protocol development and writing a good protocol is a continuing experience. The more we keep writing protocols, the more we become better. There are a few questions, but in the interest of time, what we'll do is we'll take all these questions during the Q&A session at the end of the today's workshop. That will be around 12:45. So we'll have a quick 5-minute break, and we'll come back after 5 minutes for the next session by Dr. R.N. Pandey on biostatistics.
Okay, ma'am. Thank you.
Thank you, Dr. Satyajit.
completed 2 years of training in population studies from the International Institute of Population Sciences, Mumbai. He is a PhD from the IIT Mumbai before joining AIMS New Delhi. In 1992, as an assistant professor of biostatistics, he has worked as a biostatistician from 1984 to 88 at ICMR and 1988 to 92 at NII New Delhi. During 1995 to 96, on Rockefeller Foundation Fellowship, he has been visiting scholar and a postdoctoral fellow in biostatistics and clinical epidemiology, and a Robert Wood Johnson's clinical scholar at the UNC Chapel Hill, North Carolina, USA. For the past 14 years, he's a professor and head from the Department of Biostatistics, AIMS Delhi. Also, he is a professor in charge in Clinical Epidemiology Unit in AIMS Delhi. His areas of research include statistics aspects of both observation and experimental studies, with particular interest in non-communicable diseases. He has teaching and research experience of more than 30 years. He is a member of various scientific committee of DBT, DST, ICMR, PHFI, and AYUSH. He has more than 400 research publications in peer-reviewed medical journals. He has been a principal and a co-principal investigator in more than 20 funded research projects. He has been guide, a co-guide for more than 150 MD, MS, DM, MCH theses. For his academic contribution in the field of biostatistics, he has been awarded as a Fellow of the Royal Statistical Society, FRSS, UK, a Fellow of the Indian Society of Medical Statistics, and a Fellow of National Academy of Medical Sciences. It's our honor and our pleasure to have you here with us in this workshop, sir. The session is over to you.
Uh, Good morning, and thank you very much for the kind introduction. Let me share my screen. Is my screen visible?
Yes, sir, it's visible.
Okay. First of all, I would like to thank Dr. Melvin for having me on this workshop, and good morning to everyone.
Morning, sir.
So I have been asked to speak on basics of biostatistics. So it will be absolutely basics, right? So let us proceed. You have been hearing hearing a lot of things since yesterday. This population you have heard. Since this is workshop on research methodology, we'll be dealing with research question. Your research question is always for a defined target population, but you will never be able to study everyone in the target population. So every research question is for the identified target population, will never be study entire population. One always studies a sample from the population. Sample means not any sample, it has to be representative sample. So the sample is part of the target population. So you identify sample, you enroll sample, and on sample you collect data, right? So you also heard so many study designs. So you had observational study design, you heard experimental study design. So either you draw or you enroll study participants from the population, and on sample you collect data. You may have 100 participants, 200 participants, maybe 20,000 participants. So that's your sample size, and you collect data on them, whichever way, pro forma or tab or whichever way, and you analyze that. So you analyze that, it tells you what did you get in your sample, but that is not your objective. Your objective is to answer a research question for the target population based on sample finding. So you collect data on sample, you analyze this, and whatever result you have got on sample, that you extrapolate to your target population. So that's how research works. It starts with population, ends with population. You have uncertainty research question for population, you collect a sample, enroll a sample, collect data, analyze, and you extrapolate to the population. That's how research works. Now, research question, you had very nice session yesterday. research question. You will be surprised to know that in health sciences we have limited number of research question. One may say we have infinite number of research question. No, we have absolutely finite number of research questions. We only have 9 research question, no 10th research question. Only 9 research questions, and you heard 4 study designs. 3 you heard yesterday, 1 you heard today. Yesterday you heard observational cross-sectional case control cohort. Today you heard experimental. So only 9 research questions, and these questions are answered using one of these study designs. Same research question can be answered using any of these 4 study designs, but as said earlier, one selects study design based on whatever constraint or whatever, based on feasibility. A lot of things are there. So basically research question determines your study design. Study design determines how you enroll your participants, how you collect your data, and the way you have.
So you're gone to mute.
I don't know how.
Okay, you're fine, sir.
Okay, so let me repeat. Research question determines your study design, and study design determines how you enroll your participants and how you collect your data. And the way you have collected your data will determine what analysis you do. All sorts of research question, various study designs, everything you put it in Excel sheet, but you don't analyze in the same way. So if you give me your Excel sheet, I will ask you how did you collect your data, right? So the way you have collected your data, you will use statistical methods. The way you have collected your data, you will prepare a table. We'll come to that. So research question determines your study design. Study design determines the way you collect your data and the way you analyze. And the way you have analyzed, you draw interpretation. So this is what is a sequence for research that you have to know. Suppose you are reading somebody's paper, and you have to, before you accept that fellow's interpretation, you would like to know whether data analysis is in is proper or not. Data, so this sequence everybody has to know, whether you're doing research or you are accept, or whether you are reading somebody's finding. You see, upper diagram you saw yesterday, and lower one you saw today. Yesterday you saw observational studies. Observational studies, you have a real population. Real population means living in fixed geographical area, and then then you select a sample using sampling methods, right? And you collect data on sample and you extrapolate. Today what you heard was clinical trial. You don't have a real population. You don't have a real population. You are doing a trial in hospital. You have list of inclusion criteria, exclusion criteria, followed by consent. Then you assemble the sample, right? And collect data, and whatever data you have collected, you extrapolate to a target population, and that target population is not real population, that is hypothetical population. And this sample that you have in clinical trial is not random sample, it's a sample of convenience. So in observational, you have real population, you have random sample. In intervention studies, you have sample of convenience and you have hypothetical population. If you have studied type 2 diabetic patients, non-smoker, non-obese, then the population would be type 2 diabetic patients, non-smoker, non-obese, wherever they are. But if you have done studies in Chennai, then your population, the findings will be applicable only to Chennai population, not to New Delhi population. So this is what it is. So the clinical trials, as you heard today, those are basically sample of convenience, right? And all statistical methods that you use, they are for random sample, they are not for sample of convenience. That is why in the morning Dr. Melvin explained that you do randomization for 3 reasons. One, to prevent allocation bias. Number two, if you do randomization, then you're likely to have, not guarantee, you're likely to have known or unknown confounders equally distributed. And third, for statistical reason, it gives you green signal to use a statistical method. Uh, so those are the 3 reasons so that you have to do. Now this is what is the basic. Population, your research question is for population. For your research question, the population, the answer that is there in the population is known as parameter, which is truth, which is unknown. And that's what we want to know. So to know this truth, we take a sample, and on sample, whatever we collect, whatever data we collect, when we summarize that, that is statistic. My question is prevalence of diabetes in the population. So there would be some prevalence of diabetes in the population, but I do not know. Nobody knows, right? So to know that, I will take a sample of, let's say, 1,000 individuals, and then I will find out what percentage of them they are diabetic. That is what is statistic. So statistic is nothing but the summary that you calculate from sample, and you don't calculate one thing, you calculate several things. So that is why statistics. And when you do these activities in health sciences, you call it biostatistics. Same thing you do in agricultural statistics, economic statistics, industrial statistics, and so on. So statistic is a single estimate that you have derived from the sample, and it is an estimate of the parameter. Now you can draw n number of samples from the same population, and each of the samples will not be the same because there is variability in the population. So each of the sample will not give you same result, even though everything is same. Your sampling method is same, everything is same, measurements are techniques are same. Still, you will not get the same statistics, same result across the repeated samples, and that variation is because of sampling variation, because of being different samples, right? So whenever you do a study, you want to know what is the value, what is the parameter. You want to make a statement about the truth based on the sample finding, right? But when you report your finding, when you report your finding, two questions will be asked for your every result that you report in a study. They will ask you how valid your results are. That means what they're asking is how close your reported result is to the true unknown result in the population. Accuracy. So he will ask about accuracy. And second thing that he will ask you is, you have done only one sample. You have studied only one sample. How sure you are that your findings are reproducible? So second question is asking about reproducibility, reliability, precision, consistency, right? So these two questions will have to be answered for every research question. And when you develop your protocol, your everything that you have in the protocol is built around protecting the validity and reliability of the study. So your protocol must contain every aspect which can threaten validity and reliability of your study result. If it does not include, that's an incomplete protocol. Now, so what is so for somebody who is working in health sciences, he should know three things. He should know three things to say that he knows adequate biostatistics. One is, of course, he should know a little bit of statistical methods. He should know research methods, and then he should have computing skills. If you have all the three, then you can say, yes, I know biostatistics. So a biostatistician should be comfortable in each of these three things. So what are the statistical tools? Statistical tools, basically, they help you in providing valid and reliable results. So let us go to variable. Research deals with variability. No variability, no research. This is the bottom line. Research deals with variability, no variability, no scope for research, right? So in this section, we'll talk about variables, and you need to understand variables from two perspectives. You have heard these terms. You heard the term outcome, exposure, other factors, right? You also heard the term, it's just previous session, you heard scale of measurement, right? So we need to understand variables from two perspectives. One, from their functional relationship point of view. What is the relationship in them from research question point of view? Second is, how are you measuring them and how are you analyzing them? These are the two things will determine your statistical methods. So understanding of this is absolutely essential. If you are attending any workshop on research methodology, these are the ABCD of research methodology. Whether you know, remember anything or not, you have to remember these five terms: outcome variable, exposure variable, confounding variables, bias, and chance factor. Research deals with variability, and every researcher deals with only three kinds of variable, no fourth kind of variable. Absolutely no, there is no fourth category of variable. There are only three category of variables, right? Outcome variable, exposure variable, and other factors. Outcome variable, exposure variable, and other factors, these are the variables. So if I see your protocol, then I should see what are the outcome variables, then I should know what are the exposure variables, then I should know what are the other factors, what are the confounding variables, right? So one collects data on variables and only these three types of variable. Then other two things you have is bias and chance factor. So these are five terms that you have to remember: outcome variable, exposure variable, confounding variable, bias and chance factor. Outcome variable, it was very nicely defined yesterday. Uh, Outcome variable is something in which you are actually interested in, right? Outcome is not, outcome does not mean only that you give something and some effect happens. Of course, that is one kind of outcome we are interested in. But suppose I'm interested in knowing what percentage of school children they are smokers. So smoking is the outcome variable. Anything in which I'm interested in, that is outcome variable, is known by different names. Some people call it outcome, dependent, effect, response. And so suppose somebody says I'm interested in smoking versus hypertension. So hypertension is the outcome, right? Smoking would be the exposure. But smoking is not the only exposure which can influence hypertension. There are a whole lot of other factors which can influence hypertension: obesity, family history, poor nutrition, uh, physical inactivity. So many factors are there. Many of them we don't know. So those are other factors. Those are other factors. Second person says, actually, I'm interested in hypertension versus CVD. So for him, hypertension becomes the exposure. So what is exposure? What is outcome depends on your research question, right? So suppose my research question is smoking versus hypertension. So when I prepare my protocol, I have to think of other factors also, right? I have to think of other factors. Any outcome you take, whether in non-communicable diseases or communicable diseases, there is no single exposure. But in a given study, researcher is interested in knowing role of an exposure on an outcome. Everything else or all other factors, they become villains, they become nuisance, and they will not let you know the true relationship between your exposure of interest and outcome. So those are other factors, right? So other factors you have to anticipate at the protocol writing level. You have to collect data on them. I will show you the last slide where the possibility for associations are four, that you will see later. So outcome variable is every study will have outcome variable. You cannot say in my study I don't have outcome variable. That means you are saying that I'm not interested in anything. Then why are you doing research? Every study has to have outcome variable, whether descriptive or analytic. Exposure variable is any factor which has potential to influence the outcome. It is also known by different name: exposure independent covariate factor. So these are basically exposure variables, right? This exposure variables will have n number of exposure variables, and out of them you will be interested in knowing role of one exposure on one outcome, like smoking versus hypertension, right? So these exposure variables, again, you can divide into two categories: modifiable, non-modifiable. Like age, gender, you can't modify. You can't, if you do a study for 5 years and say age and gender are the biggest predictors or biggest risk factor, you can't modify them, right? So in research, our focus is on identifying modifiable risk factors, right? But you may say environment is modifiable, but that's modifiable, but researcher cannot modify. So those are modifiable. But then other type of variables are intervening variables, right? Intervening variables are, they will be in between exposure and outcome. Suppose I am interested in knowing relationship between alcohol consumption and MI. So blood pressure, BMI, lipid parameters, they would be intermediate. Like alcohol consumption influences BP, and BP is a known predictor of MI. Alcohol consumption influences BMI, non-predict. So these are intermediate outcomes. They are not confounders, right? So many papers you would see that people have adjusted for intermediate outcome, which is wrong. If you adjust for intermediate outcome, then the relationship between exposure and outcome moves towards null. So that, please be careful with intermediate outcome. So variables you have to understand from their functional relationship point of view, that is outcome, what is exposure, what is other factors. So if you come to me for analysis, I will ask you what is outcome, what is exposure variable, and what are the other factors. Then I know what analysis to be done. Second thing is, these variables have to be actually measured. You have to collect data on them. So you collect data on them either in categorical form or in quantitative form. Suppose your outcome is cure. So cure is yes or no, categorical. Or suppose your outcome is, let's say, birth weight of newborn. So that is quantitative, right? Or your outcome is BMI. So that is quantitative. or same BMI you can analyze as obese, non-obese, so categorical. So your outcome can be categorical or quantitative. Your exposure can be categorical or quantitative. Your other factors can be, some of them can be categorical, some of them can be quantitative. So we measure these variables either in categorical form or in quantitative form. Every variable has its level of measurement, and when you collect data on them, you should try to collect data on them at highest level of measurement. For example, age. Do not collect data on age in categorical form, 10 to 20, 21 to 30. No, collect in quantitative categories. You can create anytime. So these are the so variables you have to understand from two perspective. One is relationship point of view, what is outcome, what is exposure, what are the other factors. And second is when you're analyzing, when you're collecting your data, in which form you're collecting, and when you're analyzing, in which form you're analyzing. So choice of statistical method depends on the form of variable at the stage of analysis, right? Like hypertension is not a measured variable, it's a derived variable. BMI is not a measured variable, it's a derived variable. Derived means you are creating this variable using some of the measured variables. Like hypertension, you need systolic blood pressure, you need diastolic blood pressure, you need another variable, whether person is on any antihypertensive medicine or not. So combining these three, you create hypertension. Same way BMI, you need weight and height, and then combine and get BMI. So these are the variables. So variables you need to understand from two points of view: one from relationship, second from measurement point of view. This is the basic. You need to know how to enter your data. Most of you will have you will be dealing with small data set, and you will be entering your data in Excel. So please remember, because you're going to analyze your data using some software. Any software you use, they will softwares will accept data entered in Excel. So be careful how to enter your data, right? Your rows have to be records, column have to be variable, and in any column you can enter either as alphabet or as numeric. So this is, below is wrong way of entering, above is right way of entering. So age, you can't say 30 years, it has to be 30. Then you say, no, after I want to remember after 10 years that it was in years. So write here age YRS. Similarly, gender, you, you can't say capital M, small m, and all that. It is case sensitive. So either write capital M or a small m, right? So those things you have to do. Lost, you cannot say either number or alphabet. And another rule is the data for the first participant has to be in row number 2. You cannot take more than one row for name of the variable, right? So data, so this is another wrong way of entering it. So these are basic rules for entering your data, so that just remember. Now, let us assume that you have collected your data, you defined your research question, study design, all those kinds of things. You collected your data, you prepared your data. Now you come to basic analysis. Now, analysis, there are only two methods of analysis. One is you tell someone what is there in your sample. You have collected data on sample, you describe what is there in your sample. And the methods used to do that, they are descriptive methods. But that is not the objective of your study. Your objective of your study is, based on what you get on descriptive methods, what can you say about the target population? So you are supposed to infer your statistic, you are supposed to infer your sample results to target populations. And method used to do that, they come under the category of inferential methods, right? So there are only two kinds of methods. There are only two ways of analyzing your data. Two types of methods we have. One is descriptive, second is inferential. The results that you get out of descriptive methods, they are absolutely 100. You have to be sure about that. No uncertainty. If your sample size is 100 and mean BMI, if you say 26.4, and if I ask you how sure you are about this, then you have to say I'm 100% sure. Because if you say I'm not 100% sure, that means there is something wrong with your measurement. So descriptive methods, you have to stand by your results, but the same BMI, when you extrapolate to the entire population, your sample size is 100, your population size is 1000, and when you extrapolate the finding of 100 to 1000, that means you are applying the results of 100 also to non-sample 900, whom you have not seen, and they may be different than what you have seen. So inferential is never certain. Inferential is never certain, it is always uncertain. So when you extrapolate your sample finding to target population, one will ask you, tell me how uncertain you are, or tell me how uncertain you are about what you are saying. That's where you use statistical tests and you generate hypothesis, and you use statistical tests and report p-value. So they will ask you two questions. Tell me how unsure you are about what you're saying. And second thing they will ask you is, please tell me how sure you are about what you're saying. And that's where you report in, interval estimates, confidence interval, right? So when you extrapolate your sample finding to target population, two questions will be asked. Please quantify your uncertainty, and please quantify your certainty. So those are the methods they come under inferential statistics. So to quantify uncertainty, you use various statistical tests. We'll be discussing that later. And to quantify certainty, you use interval estimate, right? So let us discuss these three things very quickly. Descriptive method. Descriptive method, that means you are analyzing your sample data, and whatever method you use, and as long as you say this is what I got in my sample that I study, you don't cross your sample. That is basically descriptive. You're describing your master chart. Describing your master chart is an art. Why is it art? Because when you submit your manuscript or report, you cannot attach your master chart. So a reader should be able to visualize your master chart without seeing your master chart, with the help of tables, diagrams, and summary that you provide. So this is not a simple thing, right? So let us very quickly go to these tables, diagrams, and summary. Tables, there are basic rules for making tables. There are table manners, right? Like for dining table, there are table manners. In the same way, for making table for your manuscript or your thesis, there are manners, right? And how do you learn these manners? Look at the published papers, observe other people, see how they have done, right? Most important thing is table. Table should be self-explanatory, right? Say when I'm reading your table, I should not be turning pages to see what it is. So it should be self-explanatory. Should always have table number and title. Units must be there. Like you're writing BMI, please write unit. If you're writing age, please write unit. Is it years, months, or days? Whatever it is, because it has to be self-sufficient. You can't say on page number 14, I have already said age in years. No, it has to be self-explanatory. Again, choice of row and column as per the study design. You have 4 study designs, so you don't make tables in the same manner for every study design. No. Rule of thumb, there is a rule of thumb. All rule of thumb is, for all prospective studies, column should be exposure, and for all non-prospective studies, outcome should be exposure. I'm repeating, rule of thumb. For all prospective studies, column should be exposure, and for all non-prospective studies, outcome should be exposure. So which are the prospective studies? Cohort studies prospective, clinical trial is prospective, even nested case control that you yesterday saw is prospective, case cohort that you saw is prospective, even retrospective prospective, that also is prospective, right? So for these study designs, you have to have outcome exposure in column, and calculate percentage column wise. Always calculate percentage column wise, not row wise, right? So keep exposure in column for prospective studies, and calculate percentage column wise. Which are non-prospective? Cross sectional is non-prospective, case control is non-prospective, diagnostic test is non-prospective. So for them, your outcome should be in the column, right? So for diagnostic test, your gold standard has to be in the column, your test has to be in row. So these are rule of thumbs. Numbers, when you calculate percentage, it should add to 100. Also, very many times when we see the table, people report percentage up to 2 points. The moment I see percentage up to 2 points, then I know who you are, then I know how careless you are. If you say 46.843% are males, what does it makes no sense, right? It makes no sense. So never report percentage more than 1 digit after decimal place, and make sure that all percentages should add to 100. It should not be 99.99. 99.9, it should add to 100. So make sure those things are there. Table and text could, or okay, for binary variable, one category and all that is fine. For quantitative variable, you can, in a table you specify calculations, and for every calculation as a reader I must know what is the n behind this. If you just say 20%, then I don't know, is it 2 out of 10 or is it 20 out of 100, right? Both are 20%, but I will take 20, these two percentages in different manner. For every calculation, n must be there. Age may be for everybody, gender may be for everybody, height, weight may be for everybody, but lipid parameters may not be for everybody, right? So n must be there for every table. Same way for making diagrams. Same way for making diagram. A picture is worth 1,000 words. A picture is worth 1,000 words, right? So make diagram not to show profile of your study subject. Make diagram not to show profile of your study subject. Okay, this is pie diagram, male are these, female are these. Will anybody have any difficulty in understanding male, how many male female are there? So don't make so this kind of stupid things, right? So people in thesis, they use diagrams to show their study profile, but in published papers they use results to show, they use diagrams to show their study results. Whatever you see pictorially, that is remembered for very long. Who wants to remember the profile of your study subjects? But I would like to remember, I would like somebody to remember my study findings for for very long. So publications, people use diagrams to show their study results, but students, residents, they use diagrams to show their the study profile. Also, when you write your thesis, please don't make this kind of mistake that one table, male these many, female these many, second there's a pie diagram, and third you write a sentence. You are saying the same thing in three different ways, so one knows what kind of person you are, right? So don't expose yourself, and be very careful. Types of the graphs, it is basically, if it is categorical, pie diagram, bar diagram, I'll not get into detail. Then if it is quantitative, dot plot, histogram, stem, all that graphs are there. What are the good diagrams? Good diagrams are the ones, good diagrams are not the ones which are very colorful, which are very complicated. The basic purpose for making diagram is to convey a complicated thing in a simple manner. So if you have complicated your diagram, basic purpose of making diagram is lost. So make your diagram as simple as possible, and as long as you have been able to convey what you wanted to convey, is a good diagram. Also, see, when you make your table, please write everything very clearly. Also, the bottom of the table is very important. Bottom of the table is very important. So don't write, suppose BMI is your variable, so variable is a BMI, and in the bottom of the table you say BMI is equal to body mass index. That is not right, because you are wasting your space. Use your bottom space only to highlight important points. So if there is some result which you want that nobody should miss, so you can put a star or ABC, whatever, and then you explain below what is A, what is star. So bottom is very important. Don't waste that paper. Don't. Now now coming to the data analysis, there are only three ways one can do the data analysis. You analyze one column of your Excel sheet at a time. That is univariable. So age is a variable. So I want to analyze what is mean age, what is standard deviation, what is range. So I'm doing univariable. That means to analyze, I don't need second column. Second type of analysis are bivariate, where we are using two columns at a time, right? Bivariate. And third type of analysis is when we are using more than two columns at a time. There is no fourth way of analysis, only three ways of analysis. So in research, everything is limited. That's a good news. Everything is limited. Your research questions are limited, your variables are limited, your statistical methods are limited, your the way you analyze your data is limited. Everything is limited. So these, now when you come to bivariable, variables can be categorical or quantitative, right? So you can have four combinations: categorical quantitative, or quantitative categorical, both are same. That's why. And third is both are quantitative. So suppose if I want to know what is the relationship between let's say obesity and diabetes, right? So what I do is, obesity I take BMI, diabetes I take blood sugar. as the parameter. So I can study the relationship in four different ways. One is I take both BMI and blood sugar quantitative. So I will do correlation analysis. Second can be that I take BMI as categories and blood sugar as quantitative. So I compare mean blood sugar across the categories of BMI. Or what I can do is I can categorize blood sugar and compare mean BMI across. So method would be the same. That means I'm comparing mean across the category of whatever. And third option is that I categorize both. So I have 2 by 2 table or 2 by 3 or whatever contingency table. So these are only three ways, right? So one would do univariate, bivariate, and analysis and multivariate. In some situation you do only univariate. In some situation you do univariate and bivariate. In some situation you do all three. It depends on your situation and question. Now let's go to each of these very quickly. Univariate and bivariate. Univariate, if your variable is categorical, you will do ratio, proportion, rate, prevalence, incidence, and those kinds of things you will do. For categorical, categorical is easy. If your variable is quantitative, then it's slightly difficult. If your variable is quantitative, then you want to know the central value, you want to know the average, you want to know the average. And there are three kinds of averages: arithmetic mean, geometric mean, average is the central point. So mean, median, mode, these are the averages. Mean also, you have three kinds of mean: arithmetic mean, geometric mean, harmonic mean. So you want to know the average. Average is what? Average is representative, because you are not giving raw data to the reader, so he wants to know what is the representative, right? So you give the average, right? So your arithmetic mean, geometric mean, harmonic mean. Also the distribution of variable, depending on that you use arithmetic mean or geometric mean. So mean, median, mode, you have already, you know all this. So these are averages. But why averages are not sufficient? These are averages. Then you have location, you have measures of location, you have quartiles, three quartiles, 9 deciles, 100 percentiles. Quartiles will be three, not four. Because if you want to, let's say if you want to cut this pen into four equal parts, so you make three cuts, and those three cuts are quartiles. You divide into 10 equal parts, you make 9 cuts, those are 9 deciles. If you want to divide into 100 equal parts, you make 99 cuts, those are 100 percentiles. So these are measures of locations. So these things, why these averages or measures of central tendency, see, is not sufficient. Suppose I tell you I have five numbers. I have five numbers. Uh, mean is 10, right? So these five numbers, mean 10, that means total is 50. Now total 50 from five numbers can come in variety of ways. It can be all five values are 10, 10, 10, or it can be 8, 9, 10, 11, 12, or it can be 6, 8, 10, 12, 14. All of these total will be 50. So if I tell you n is 5, mean is 10, you cannot visualize what kind of data I have, right? So therefore, just we also need a measure of dispersion, which tells you about how dispersed the values are from your central value. So we have several measures of dispersion: range, interquartile range, mean deviation, standard deviation, right? Good statistic is the one which is based on all the observations. Range, if I have 10 numbers, range would be based on just minimum and maximum. In between values are ignored. Interquartile range, not all values are used. Mean deviation, yes, all values are used. Suppose I have 10 numbers, there will be mean. So what is the difference of first value from mean, second value from mean, third value from mean? And for all of these differences, I calculate average, ignoring the sign, ignoring the sign. That means taking absolute value. That is what is mean deviation. But this another way of getting rid of negative sign is square it. So when you square and then take the average and take the square root, that is the standard deviation, which is most commonly used. So these are, so for your variable in the sample, you have to report what is the extent of variation. And if you want to compare a variation between two groups, you use coefficient of variation, because your standard deviation is with respect to the mean. Now you can have two groups but different means, so you can't compare standard deviation straight away. Now for sampling distribution statistic, that is standard error. Suppose you do a study, you get one result. Repeat the study second time, you get second result. Repeat the study third time, third result, and so on. If you repeat 30 times, you get 30 results, right? And the distribution of, and you get 30 results. And the average of this would be very close to the true value, and the standard deviation of this will be standard error. So standard error tells you about variability of results. Standard error tells you variability of results, and standard deviation tells you variability of observations in your sample. Right, so that is what, so standard error is a measure of inferential statistics. Anything when you cross your sample becomes measure of inferential statistics. So for a standard deviation you're not crossing. So now let us go to bivariate. Bivariate, three situations, both can be quantitative, you use scatter plot or correlation coefficient. Both categorical, you make contingency table, calculate percentage and various other summary measures, odds ratio, risk difference, number needed to treat, and all sorts of things you calculate. If categorical quantitative, you compare mean or median depending on the nature of the distribution. So you do these kinds of bivariate analysis. Now very quickly let us go through some of them. Correlation coefficient, basically you make a scatter plot. You can have positive relationship, you can have negative relationship, you can have no relationship looking at the pattern of the diagram. So all this you use either Pearson correlation or Spearman's rank correlation. You have two quantitative variables. If both of them are normal, both of them are normally distributed, then you use Pearson correlation coefficient. If one or both of them are non-normal, then you use Spearman's rank correlation. Right, you convert your observations into rank and calculate the correlation coefficient between the two. When normal, when non-normal, we will come to that later. How do you decide your variable is normal or non-normal? Right, now come to the second one. So there are two kinds of measures that we use. These are the measures of association. One is absolute and second is relative. Anything, what is absolute measure? Absolute measure means you are subtracting something from something, and relative measure means you are dividing something by something. So absolute measure, the null value is 0, and relative measure, null value is 1. If you take two groups and you compare mean between the two groups, if there is no difference in the two means, then you will get 0. That is what is null. If you get two groups, if you have two groups and you are taking risk in one group, risk in the other group, and both the risks are same, then the ratio has to be 1. That is a null value. So null value for relative measure is 1, and null value for risk difference for absolute is basically 0. Risk, the absolute measures, what do they tell you? Absolute measures, they tell you excess. They tell you about excess in absolute terms. And relative things, what they tell you? They tell you excess in relative terms. So that's what is the difference in the two. Now for this, let us go to this relative measures. We use odds ratio, risk ratio, probability. So I hope you understand that odds and probability, they are not same. Odds and probability, they are not same. Let us take this whole thing as 4. Black is disease, which is 1, and white is 3, that is non-disease. So if I ask you what is the probability of a person having the disease, so you will say 1/4. That's what is the probability, right? If I ask you what is the probability of a person not having the disease, so then you would say 3/4. 3/4. So if I divide the two probabilities, probability of a person having the disease, that means 1/4, 1/4 divided by probability of a person not having the disease, that is 3/4. So 4 and 4 gets cancelled, and that's what is the odds. Odds is the ratio of two probabilities, event happening divided by event not happening, right? So these odds is basically used uh, in in race. Suppose if you go for a go for a race, and you, so which horse you will pick up? Which horse you will pick up? If a horse has participated in a race 10 times, 5 times he has won and 5 times he has lost, so it is 5/5, that is 1, right? So you use odds. So odds and probability, they are not the same. Odds will be more than the probability, because numerator is same, denominator is smaller in case of odds. So this is what it is. So odds ratio, you have this 2/2 table, A, B, C, D. So if so in this table, how many are cases? A plus C. How many are controls? B plus D. How many are exposed? A plus B. How many are unexposed? C plus D. What is A? A is cases who are exposed. B is controls who are exposed, right? So if I ask you what is the odds of being a case among exposed, so it will be A upon B. What is the odds of being a case among unexposed? It will be C by D. So A by B, C by D, AD by BC, that is what is the odds ratio. This is example. And since this is ratio 3.8, right? 3.8, you can also do AD by BC. So 3.8. 3.8 means what? The odds of having MI is 3.8 times, not 3.8 more. 3 point, because this is a ratio measure. This is a ratio measure. This is 3.8 times more for smokers as compared to non-smokers. Now this is a risk factor. More than your outcome is bad, your outcome is bad, your exposure is bad. Your outcome is bad, exposure is bad. So more than one will be a risk factor, right? Suppose if your outcome is good, suppose your outcome is good, if outcome is good, when you, so now suppose your exposure is good, your exposure is good, let's say physical activity yes, physical activity no, then your odds ratio will be less than 1. That will be, so this will be a protective factor. So more than 1 is a risk factor. And again you have to see your factor is a good factor or a bad factor. So accordingly you decide. Risk difference we have already seen. Risk in one minus risk in two, that is excess risk in absolute terms. Take this example. So what is the risk of MI among those who are oral contraceptive users? That is 13 upon 5,000. What is risk in the other group? 7 upon 1,000. So the difference is 0.0019. So you can say 19 women per 10,000 can be prevented if you withdraw this bad exposure. That is what. So risk difference is basically can be used as an indicator for effectiveness for different types of treatment. 1 upon risk difference is also number needed to treat, that is in intervention studies. By the way, 4 study designs of cohort studies and clinical trial, that is prospective, case control and cohort, case control and cross sectional, they are non prospective. There are 9 research questions, 4 study designs, and only 2 ways of analyzing your study. Cohort study and clinical trial and analyze in exactly same manner. Cohort studies and clinical trial are analyzed in exactly same manner. There is no difference between cohort study and clinical trial except one that allocation is being assigned by the investigator. So method of analysis is exactly same in prospective studies, and method of analysis is exactly same in your non-prospective. So you use odds ratio, prevalence, association. You use the word prevalence, association, and odds ratio in non-prospective. And you use the word incidence, not the prevalence. You use the word incidence, you use the word predictor, and you use measure risk ratio, risk difference in prospective studies. So these three are, so method of analysis only two. One for prospective, other one for non-prospective. Right? So relative risk, I'm going a bit fast because I have some more slides. Relative risk, again, risk, this tells you in relative terms. This I already explained to you. Since this is a ratio measure, one implies rates are equal, null, right? And more than one is protective. So that I have already said. This is another example. Same way you can calculate. I will just skip. I will skip this part. So basically summary measures, summary measures. So what are the summary measures? There are summary measures: odds ratio, risk difference, number needed to treat. These are when your outcome is categorical. I have not, see, I told you there are only two kinds of outcome variable. Third one I have deliberately, I have not told you. Third one is the combination of the two. That is basically time to event. And whenever your outcome is time to event, the summary measure that you use is hazard rate. That's where you use the survival curve and Kaplan-Meier, log rank, and Cox regression. Those are the methods which are used when your outcome is time to event. And when do you consider time to event? When you assign importance to the time at which event occurs. Like cure at 6 months and cure at 2 years, both are cures, but they are not same cure. Death at 6 months and death at 2 years, both are deaths. But they are not same deaths. So when you assign importance to time at which event occurs, then you use time to event, and that's basically categorical. Status is categorical and time is quantitative. So that part I have not mentioned to you. So we discussed descriptive, odds ratio, risk difference, whatever you calculate, they're all descriptive. Whatever you calculate, they're all descriptive. Now let us take inferential. In inferential, you have this estimation, point estimate, and interval. Point estimate is nothing but what you get from your sample. So that is nothing but descriptive statistics. So this is what is the link between descriptive and inferential. Interval estimate is basically confidence interval. So confidence interval is basic. See, a reader, a reviewer does not know what is the true result. He does not know what is the true result. So therefore, what he wants to know from you is, what did you get in your study? What result did you get in your study? And what kind of results you're likely to get when you repeat your study 100 times. So please tell me range of results that you're likely to get 95% of times. That is what is your 95% confidence interval. And that is what answers the two question, that is reliability, right? Reliability is nothing but reproducibility. That's nothing but precision. That is nothing but confidence interval, right? So if you have not reported confidence interval for your primary outcome or secondary outcome, then you have not answered the second part. Again, I'm repeating, two questions will be asked: validity and reliability. Validity is the function of conduct of study. Validity is the function of conduct of study. You can ensure validity by doing your study well, measurements, analysis properly, adjustment, all that. Reliability, you cannot, reliability is a function of sample size. Reliability is a function of reproducibility, is a function of sample size. By increasing your sample size, you cannot make your study more valid. You have population of 1,000 individuals, and if you study entire 1,000 population in a wrong manner, you get wrong result, not valid. But the same population you study 10 times, you get the same result every time, wrong result. You're absolutely reproducible, right? So reliability is for, validity is the priority in research, not the reliability. Validity is the priority in research. So pay more attention to design, analysis. Sample size is basically that determines the reproducibility. So come to the confidence interval, statistical point estimate, interval estimate. Point estimate for what? Mean, proportion, odds ratio. When you do your study, you collect data on so many variables. Some of the variables are background characteristics like age, gender. You will not calculate confidence interval for mean age. You will not calculate confidence interval for percentage males, because that is not part of your objective. Your objective is not to estimate mean. So confidence interval has to be calculated for whatever is there in your objectives, whatever results that you have, right? Your results can be mean, proportion, odds ratio, correlation coefficient, and for these you calculate your interval estimate, which is confidence interval, right? So 9 questions I have been saying. What are the 9 questions? First question is prevalence. Somebody is interested in estimating prevalence. Second question is somebody is interested in estimating mean. Third question is somebody is interested in knowing correlation coefficient between two parameters. Fourth question is somebody is interested in knowing agreement between two methods or two raters. And fifth question is one wants to evaluate diagnostic characteristics of a candidate test with a reference. 5 questions. If your question is one of these five, your study design is cross-sectional. So what are five? Prevalence, mean, correlation, agreement, diagnostic test. So if your question is one of these, study design is cross-sectional. Then another 2 question. What is incidence of an outcome? What are the predictors of an outcome? Study design is cohort. So 5 plus 2, 7. What are the risk factors of an outcome? That is case control. That is number 8. And number 9 is your clinical trial. You want to, so 1 to 7 is observational, and number 1 to 8 is observational, and number 9 is your intervention, which is clinical trial. Clinical trial, you have 36 variations of clinical trial. 36 variations of clinical trial. So if you take 4 features, you heard in the morning, your trial by design can be parallel or crossover. You heard that crossover. By randomization, it can be unit randomization or cluster randomization, right? So 2 into 2, 4. Now your trial, the you can be by effect, it can be superiority or equivalence or non-inferiority. So 4 into 3, 12. Now in each of the trial, your outcome can be either quantitative or categorical or time to event. So 12 into 3, 36. So there are for a statistician, there are 36 variations of clinical trial, sample size calculation and analysis point of view. And each of them, you calculate your result and confidence interval. right? So this is what is sampling distribution of mean. So basically these are the repeated samples, and you get distribution like this, and it will have properties of normal distribution, and you calculate confidence interval. Now let me, almost always you calculate 95% confidence interval, but if you require you can calculate 99% confidence interval. For 99%, this is the multiplier. So let me go to this. Just let me give an example. In a study of a sample of 100 subjects, it was found that mean systolic blood pressure was this, SD was this. So calculate a standard error, you will have two values, and these are the confidence interval. You calculate your confidence interval for the mean and report. This is for percentage. So you can calculate confidence interval for odds ratio, risk ratio, difference in mean, whatever it is. You can calculate correlation coefficient, whatever you can calculate, whatever result you can calculate from one sample, you can calculate from repeated sample, and therefore you can have 95% confidence interval. This is another interpretation of confidence interval. These are 20 studies. These dots are the point estimate, and these lines are the confidence interval. True value in the population is 24.2. So out of this 20, 95% of the studies, which is 19, will capture the true value. One will miss. So this is what is interpretation of confidence interval. Now, very quickly, let me come to the hypothesis testing. I'm sorry. Can I take another 10 minutes, please?
Yes, sir. Please go ahead.
Okay. So let me come to the hypothesis testing. Now, hypothesis, which studies, yesterday also you heard about this hypothesis, no hypothesis. There are only two kinds of studies. There are two kinds of studies. One where you are not comparing anything, and second type where you are comparing something. So wherever you are comparing, those studies are called analytic studies. And whenever you are not comparing, those are called descriptive studies, right? So hypothesis will be only in analytic studies, right? Like if you want to know what is the prevalence, so there's no hypothesis. But if I want to know, is the prevalence of obesity same in boys and girls in schools, then I'm comparing, right? That's a hypothesis. Even in prevalence studies, even in cross sectional studies, single sample, it can be analytic. For example, somebody did a study, let's say 5 years back, among school children, and let's say prevalence of dental caries was 40%, right? Now the last 5 years something happened, some good things happened, and now he wants to know whether the prevalence has remained same or not. So he will do a survey, find out the prevalence, and he will test this prevalence against hypothesized value of 40%. So this will be analytic. Now whenever you calculate sample size for analytic, you will have power also in addition to alpha. Whenever you calculate sample size for descriptive studies, no power, only precision. So that is why you see 4 pq upon d square, there is no beta, because that's not analytic study. But when you're comparing two means or two proportions, then there is a basically beta also will come. So those things are when you for sample size. So hypothesis. So let's go to hypothesis testing. Null hypothesis, alternate hypothesis. So null hypothesis, alternate hypothesis are not your hypothesis. These are statisticians' hypothesis. Your hypothesis is research hypothesis. Your hypothesis is research hypothesis. Null hypothesis is not your hypothesis. That is statistician's hypothesis. With, can you please? Okay. So with research hypothesis, you have collected your, please mute yourself. With research hypothesis, you have collected your data, and then you come to me saying that, look, this was my research hypothesis, and this is the limited evidence data that I have. Can you please tell me how compatible the two are? So at this stage, now I'll formulate my own hypothesis, right? Why? Because it is easy to disprove something than to prove something. So null hypothesis is my working hypothesis. If I say all cars in Chennai are red, right? So to prove this, I have to see all the cars. But to disprove that, the moment I see non-red car, it is disproved, right? So it's easy to disprove something than to prove something. So that is why null hypothesis becomes my working hypothesis. In hypothesis testing, the procedure that we follow is 100% same as what happens in our judicial system. Judge is the biostatistician, and police police is the investigator, and they take they take a fellow to the judge with limited evidence. So when it goes to judge, so he formulates his null hypothesis, and the null hypothesis research. What is research question? Research question is the person guilty or not? That was research question. And what is the research hypothesis? Person is guilty. What is null hypothesis? Person is not guilty. Right? So judge formulates his null hypothesis. That is the law of the land, that a person is innocent till proven guilty. So his null hypothesis is person is innocent, and then he wants to see how compatible the limited evidence are with the person being innocent. He tries to reject this. If he is unable to reject this, then he declares that okay, person is guilty. But even when he says person is guilty, he cannot be 100% sure, because he's dealing with he's dealing with limited sample, limited data, right? So there will be uncertainty, and that is what is the p-value, right? That is what is the p-value. So that is what there is a process of hypothesis testing. So I would formulate null hypothesis and alternate hypothesis. It can be one. So those are things that I will say, right? Your hypothesis can be one-sided, can be two-sided. Now, almost 95% of the situations, it is two-sided. It is always, it is 95% times, it is two-sided. For your alternate hypothesis to be one-sided, you have to be really sure that it is not possible to get in other directions. Now, if it has implication, one-sided, the sample size is less, but not half, because the multiplier for one-sided is actually 1.64, and multiplier for the two-sided is 1.96. It comes in the numerator. So difference is not exactly 50%, but the choice of one-sided or two-sided should not be that in one-sided I get less sample size, so let me take one-sided. No, it has to be based on your scientific rationale. These are the two errors. Now, whenever you are taking a decision, some amount of error is likely to happen. Whether making a diagnosis, you are likely to make it like a error. You are going to buy a TV, you are likely to make error. You are going to buy mobile phone, you are likely to make a error. Any stage where you are in a you have to take a decision, you are in a 2 by 2 table. You are always in a 2 by 2 table, right? Because reality can be like, you are, as a doctor, you are looking at a patient, he is presenting to you with limited symptoms, and that many times overlapping symptoms, and you have to take a decision, disease or no disease, right? So again, you are in a 2 by 2 table. You have true positive, false negative, true negative, right? So same way in research, you're likely to make mistake. You're dealing with sample. The tools that you have are not 100% sensitive, specific. Even when you're asking question, you're not sure whether the fellow is telling 100% correct or not. So some amount of error will happen. So that's where is false positive and false negative. And the rate at which you commit false positive is type 1 error, and the rate at which you commit type 2 is is basically type 2 error, false negative. So for a judge, which error is more serious? For a judge, he says, let many guilty go scot free, not too many innocent should be punished. That means he minimizes false positivity. And false positivity is alpha. That is why your alpha is never more than 0.05, 5%. Beta can go up to 20%, right? So these are the two errors that will happen, right? So power, all that is fine. So let me go to very quickly we go to we go to statistical test, and we will see how, what is the criteria to pick up the test. What is the criteria to decide which test I should pick up in this situation? Now take this example. You have drug A, drug B. The survival rate in drug A is 68%, and survival in drug B is 48%. So A is 20% superior than B. Now this 20% is really meaningful, clinically meaningful. Now then you should apply statistical test. So this clinically meaningful result that you have got, is it the true result or is it by chance? Is it the true result or is it by chance finding? Then you use your statistical test. Suppose this was 68% and this is 67%. So will you 1%, is it meaningful? If it is not meaningful, then why do you use statistical test, right? statistical test should be used only when you see a clinically meaningful difference, and then you would like to know that this clinically meaningful difference, is it a real difference or is it just by chance? How often this meaningful difference can come because of chance? That's what you would like to calculate, and that is what is the p value, right? So if I do statistical test between 68% and 48%, and if I get p value of zero point zero 0.01, what does it mean? I would conclude that drug A is better than drug B, because my cutoff is zero point zero 0.05. That is arbitrary, but that is what is taken. I would conclude a positive, my finding would be positive or conclusive. But again, in the bracket, I would say p is equal to 0.01. That means what I'm saying is 1% of the time, this kind of finding could occur due to chance alone. So p value is nothing but the role of chance in a conclusive finding, and that's what we do in statistical tests. So I told you three situations: categorical, 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 quantitative, quantitative, quantitative. Now, these are the 4 points that you should remember while picking up a statistical test. Which test is to be used in which situation, to know that these are the 4 points. Like every individual has his address, and address has 4 points. Dr. Melvin George, Department of Community Medicine, SRM Medical College, Chennai. That is his address. In the same way, every statistical test has 4 lines in the same order, because in Dr. Melvin's address, you will not have Chennai first and Dr. Melvin at the bottom. There is an order. So this is what is the order. First is combination of two variables. Second is your variable is normal or non-normal. And third is the groups you have two or more than two. And third is the groups that you have, are they related or unrelated. Now remember, when I said combination of two variables, there are only three possibilities: both categorical, categorical quantitative, both quantitative, right? So if I say I want to compare age between males and females, so it is basically categorical versus quantitative. If I say I want to compare smokers, I want to compare hypertensives between smoker and non-smoker, so both are categorical, right? If I say I want to see relationship between BMI and blood sugar, both are quantitative, right? So if I say I want to say BMI below 18, 18 to 25, and 25 plus, and in them I want to compare blood sugar, so basically is categorical versus quantitative. Your categorical variable BMI has got three categories. So it's bivariate. Even though you have three categories, but variables are bivariate, because two columns of Excel sheet. So you have to see. So just remember these four things. If you have understood this, then you have understood all the statistical tests. If you have understood these four, you have understood all the statistical tests. I am going to discuss 11 statistical tests with you, which are used 95% of the times. 11 statistical tests which are going to be used, which are used 95% of the times, and which test you use is determined by these four things. Let me explain to you each one of them. Combination of two variables, I have already explained to you, right? There can be three combinations: categorical, categorical, categorical quantitative, and third quantitative, quantitative. Second is normal, non-normal. Normal, non-normal would be an issue only for quantitative variable. So the three combinations that you have for categorical, categorical, this will not be an issue, because there is no quantitative. So when do you say that this variable is normal or non-normal? If your variable is normal, you use parametric methods. If your variable is non-normal, you go other way, you use parametric method. So you need to decide, when should I use parametric, when should I use non-parametric, that depending upon normal or non-normal. So there are some criteria. We'll come back to this. Third is groups two or more than two. If I'm comparing age between males and female, so my variable is gender, and male and female are the groups. So these are two groups. If I have severity of disease, mild, moderate, severe, and I'm comparing CD4 count, right? So my group variable has more than two categories, right? So I don't need to discuss two or more than two. You can figure out groups are two or more than two. Fourth is related or unrelated, right? So I would discuss normal, non-normal, and related, non-related, right? So these four things you must understand very quickly. And once you have understood these four things, then you have understood 11 statistical tests. These are the combination of two variables. First one, I have already explained to you. Then normal means if it's for quantitative variable. So if your variable distribution is like this, it is normal, like this, non-normal, right? If your see, if your variable is quantitative and if it is normal, use parametric methods. If it is non-normal, then you make some transformation. Like you saw geometric mean. What is geometric mean? Geometric mean is nothing but suppose I have three values, 10, 100,000. So I take log of this, log of this. So log of 10 is 1, 100 is 2, 1000 is 3. Now for 1, 2, 3, I calculate arithmetic mean and take anti-log, right? So 1, 2, 3 is normal, but 10, 100,000 is not normal, right? So you convert into normality. There are a whole lot of transformations, log, square root, 1 by x. There are several transformations. You have convert into normal. Not log, not same transformation will be applicable for every variable. You have to see which one is applicable for what variable. So transform into normality. Once you have transformed into normality, use parametric methods. Parametric methods are powerful methods. In parametric methods, the the values which are being used are the original values, and in non-parametric methods the values which are used for calculation are not the values that you have measured, but their corresponding ranks. So 10, 100,000, if you use non-parametric, the inputs will be 1, 2, 3, right? So non-parametric methods are supposed to be poor methods, weak methods, right? So as far as possible, you try to use parametric methods. Now, related, unrelated. Related, unrelated, simplest is before, after, pre, post. Second is you have two groups, you have matched. Suppose I want to know relationship between smoking and hypertension. I want to do a case control study. So I will take hypertensive one group, non-hypertensive other group, right? But I match them on obesity, third factor, right? So that will be matching, right? The third criteria is basically clustering. Clustering means the outcomes are not independent. Cluster does not mean set number of individuals. Even single individual can be cluster. For example, if you measure my blood pressure today, tomorrow, day after tomorrow, and 5 different days, so these 5 values are not independent. They are coming from same individual. That is cluster. So clustering is again related. So your observations can be related in variety of ways: before, after, matching, and clustering. So four things. What are the four things? Number one, combination. Number two, normal, non-normal. Normal, non-normal. Number three, groups. Number four, related. Normal I have not discussed. Normal is distribution of variable normal, not in your sample but in the population. But you don't have access to the population values. You have access to only sample values, right? So rule of thumb, textbook mentions rule of thumb. If your sample size is adequate, more than 20, calculate three things: mean, median, and standard deviation. If your sample size is more than 20, calculate mean, median, and standard deviation. If mean, median are not comparable, then look at the standard deviation. And if the standard deviation is more than 40% of the mean, if the standard deviation is more than 40% of the mean, then you take it as non-normal. And non-normal variables have to be summarized not as mean SD, but as a median and range or interquartile range. So normal variables are to be summarized by mean SD, and non-normal variables are to be summarized by median and range. I told you, if it is non-normal, you make an effort to convert into normal and use parametric methods, calculate mean. Like for example, geometric mean, right? So those are the things. Now I'll show you just 2 3 slides. Your categorical versus categorical, you have three methods: chi-square test, Fisher's exact test, McNemar test. Chi-square test you use in a contingency table where you have 2 by 2 rows or more than two, or say 3 by 3 rows. But condition is to use chi-square test. Condition is there should not be any matching. And another is that if you have a 2 by 2 table, there are 4 cells. If you have 3 by 3 table, there are 9 cells. And none of the cell frequencies should be less than 5. To use chi-square test, there are two conditions. One is there should not be any matching, and none of the cell frequencies should be less than 5. If it is less than 5, then you don't use chi-square test, use Fisher's exact test, right? And McNemar test, when do you use? When your groups are related. So there are 3 tests here. I told you I will be discussing 11 tests. So 3 are here: chi-square test, Fisher's exact test, McNamara test. You also have test of proportions, that is same as your chi-square test. There is a link between a chi-square and Z. All those statisticians would know. But these are the 3 tests. So when these are 3 tests used when you are categorical versus categorical. Now let's go to second slide. When it is categorical versus quantitative, and your parametric means normal. Parametric means normal. Groups are 2, then use students t-test. So when would you use students t-test? I would use students t-test when my groups are 2 and my quantitative variable is normal and the groups are unrelated. So to compare mean between 2 unrelated groups, I will use students t-test, right? When will you use pair t-test? When you have pre-post kind of thing related, you will use pair t-test to compare mean. Now extension of students t-test, you have group A, group B, group C. Then you say no, I will use A versus B, A versus C, and A versus B. I mean, I'll use 3 pairs. That is not allowed. So you have to do one-way ANOVA. One-way ANOVA is extension of students t-test. One-way ANOVA is extension of students t-test. In students t-test, null hypothesis will be mean 1 is equal to mean 2. In one-way ANOVA, your null hypothesis will be mean 1 is equal to mean 2 is equal to mean 3, right? So in one-way ANOVA, when your null hypothesis is rejected, that means mean 1 not equal to mean 2 not equal to mean. So all the 3 null hypothesis rejected, that means all the 3 means are not the same. So in that case, you go and do post hoc ANOVA, like you have Bonferroni, Skiffy, SIDAC, several Dunnett's, several post hoc ANOVA you have. They basically to tell you which pair or which pairs are different. So that is called post hoc ANOVA. And repeated measure ANOVA is extension of pair t-test. Like my blood pressure today, after 10 days, after 30 days. So these 3 groups, they are not independent related. So here what we are doing is we are comparing mean. In these 4 tests, we are comparing mean, but you can have the same situation where not the mean, but you are comparing the median. And that's where you have other 4 non-parametric tests. So Wilcoxon rank sum test, what are you comparing? You are comparing median between 2 unrelated groups, right? Median between 2 unrelated groups. This Wilcoxon rank sum paired. Kruskal values, basically like one-way ANOVA, but you are comparing median. You take any 2 tests. You take any 2 tests and you will find 3 things common, one different. Any 2 tests you take. Say, for example, the students t-test and pair t-test. Which one is different? Fourth one is different, related, unrelated. Other things are same. You take students t-test and Wilcoxon rank sum test. Which one is different? Second is different, normal, non-normal. Every other 3 are same, right? You take students t-test and one-way ANOVA. 3 things will be same. Which one is different? Third one, group 2 or more than 2. So you take any 2 tests, you will find 3 things common, one different, right? So in students t-test, what are you doing? You are comparing mean between 2 unrelated groups. In pair t-test, what you're doing? You are comparing difference in mean. Difference in mean. So basically you will have series before, you will have after. For every individual, find out the difference and mean difference, right? So null hypothesis. So even though it is 2, but pair t-test become single sample test, right? One-way ANOVA, you are comparing mean between more than 2 unrelated groups. Repeated measures, you are comparing mean between more than 2 related groups, right? Kruskal values, what are you doing? You are comparing median between more than 2 unrelated groups. Wilcoxon rank sum test, what you're doing? You are comparing median between, you are doing the same thing what you're doing in students t-test, but here you are comparing median because this is non-parametric, right? So 4 here and 4 3 previously. 8 plus 3, 11 statistical tests, right? So this is what is again a clinical significance. I will not get into all that. So I know I've crossed my, now this is the last slide I'm telling you. Any association that you're showing, any association that you are showing, if you tell me that I did a study to show my drug A is better than drug B, or I did a study to show smoking is associated with lung cancer, right? You showed the result that yes, my results are showing this. I will not believe this. I will not believe, nobody will believe this. They will try to find out, what all can be the what all can be the other explanation of of what this fellow has observed. So I will see, can this finding be, can bias be the possible explanation of what this fellow is saying? So it is your duty to explain to me that, look, whatever I'm saying is not because of bias. So how you will be able to say this? That means in your protocol, you should have anticipated that in what kind of different ways my study could be biased, and what did you do to make sure that they don't do it? Like, did you, for a clinical trial, you did that randomization, right? Also, you did blinding because outcome assessment bias. So all the, there are more than 50 types of biases. You have to see in my study which bias is applicable and what strategy you had to minimize bias. There is no statistical method which can take care of bias, which can correct for bias. Bias is responsibility of investigator. So that is where your method section is important. So I get convinced by your method section that minimum bias. There is no study free from bias. Every study has bias. Good studies have minimum bias. So I am okay. Okay, he has taken care of bias to whatever extent possible. But still, I will not believe. Still, I will not believe, because I know there can be other factors. There is no single factor. So you have to explain to me that whatever you are saying is not because of other factors. It's not because of other factors. So other factors, bias is not a possible explanation. Other factors are not possible explanation. Still, I will not believe, because you have studied only one sample, and this can be a lucky sample. So you have to convince to me that bias is not the possible explanation, other factors are not the possible explanation, and whatever I'm saying is beyond chance. Then only I will accept what you're saying, right? So this is what is your basically epidemiology. If you want to do research, anybody in any discipline, anyone in any discipline, if you want to be in research, read principles of epidemiology. If you have not read principles of epidemiology, forget about research. And this is what is the ABCD. I told you four terms. I told you four terms. Out. I told you four terms. Outcome. So just let me, just give me 2 minutes to just summarize. Uh, Four terms: outcome, exposure, other factors, bias, chance. That is number one. Second thing is, we deal with variability. Variability to understand from two perspectives: one from functional relationship, second from measurement and analysis point of view. From functional relationship point of view, you have outcome variable, exposure variable, and other factors. Other factors, again, inside, if you go, they can be confounder, they can be effect modifier, they can be intervening variable. Those are other. So broadly three variables, right? When you come to statistical, when you come to research, question is always for target population. You collect data on sample, and you do two kinds of statistical methods. One is describe your master chart, that is descriptive, and second is you extrapolate your sample finding to population, that is inferential. So descriptive, you have three: you have tables, you have diagrams, you have summary, right? Tables, diagrams, I told you. Summary, again, you have three kinds of summary: univariate, bivariate, and multivariate. Those are all summary, right? So you will calculate odds ratio, you will calculate adjusted odds ratio, and as long as you say this is from a sample, it remains descriptive. And the moment you add confidence interval or p value to your odds ratio, it becomes inferential. So inferential, you have two parts. You have basically estimation and your hypothesis testing. Estimation, you have point and interval, and hypothesis testing, again, you have null hypothesis and alternate hypothesis. And again, I told you four criteria which can tell you which test is to be picked up in which situation. The criteria are combination of two variables, your variable, quantitative variable, normal, non-normal. Third is groups two or more than two, and fourth is the group that you have is related or unrelated. So these are the four criteria. And for criteria number one, I told you three tests: chi-square, Fisher's exact test, McNemar test. And when you go to number two, categorical quantitative, four for parametric, four for non-parametric. For parametric, you had students t-test, pair t-test, one-way ANOVA, repeated measures ANOVA. For non-parametric, you have Wilcoxon rank sum or Mann-Whitney U-test, and Kruskal-Wallis, and Wilcoxon sign rank, and Friedman's test, right? And then for any association, these are the possible situations. So you have to think. Now, last is, don't think that biostatistics is a computing problem. Biostatistics not a computing problem, isn't it? You don't need to be good in mathematics. Biostatistics is just a statistical thinking, right? Don't fall back on statistics. It don't. I know I have a good friend. He's good in statistics. So let me be very casual with my collection of data and all that. Prevention is cost effective. Prevention is cost effective and more relevant for research. Also for biostatistics, read principles of epidemiology. If you have not read the principles of epidemiology, no, forget about it, right? So this is must. Now look at this gentleman. He has very sophisticated laptop and very sophisticated computer program. He wanted to cross the river, and he had data on depth of river at different time points, right? And what he did, he calculated arithmetic mean of the depth, mean depth, and he measured his height. His height was the more than the average depth, but he had no idea about the variation. Now he is in trouble, right? So don't depend too much on calculation. Use common sense. Okay, all the best. Thank you. If you have a question, we'll take this. Sorry,
Thank you.
sorry, for exceeding time. Sorry for exceeding time.
Sure. Thank you, sir, for your very comprehensive presentation on basic biostatistics. I understand it is definitely difficult to cover such a vast area in the short time that is given to you. But nevertheless, I think this is a good introduction for people to understand the basics of biostatistics. And from this, they can look forward to developing their knowledge and skills regarding biostatistics. So I'll just take two questions. I'll read it, sir, for you, and you could just answer the questions. In the interest of time, I'm cutting short the questions. So one is, what is the importance of coefficient of variation?
Yeah, coefficient of variation. Coefficient of variation is nothing but in the same, see, the importance is same as percentage, right? Suppose you have two groups, group A 100, group B, or group B is 50, right? And out of 100, let's say 20 have the disease, and in other group, out of 50, 20 have the disease. Now, if you want to compare the two groups, you will not ignore the denominator and say both have same number of disease, right? You have to standardize them. So 20 out of 100 is 20%, and 20 out of 50 is 40%, right? So then you can compare these two. Same thing is done for coefficient of variation. When you're comparing standard deviation between the two groups, and when the means are not the same, because standard deviation is nothing but with reference to mean. What does standard deviation tell you? Standard deviation tells you that on an average, to what extent observations are dispersed from the mean, central value. And when means are not the same, how can you compare the standard deviation? So coefficient of variation is basically, it tells you that what is the standard deviation if the mean is 100. So you standardized. That's what is coefficient of variation.
Thank you, sir. And one more question. What is the meaning of false discovery rate used in gene expression studies?
Yeah. See, false discovery rate, when you don't do, see, false discovery rate, that means false positive. False positive. So that is why, like suppose you have three groups A, B, C. You have three groups A, B, C. You did one way ANOVA to compare mean, and if you find overall there is a difference, right? There is a difference. So then A versus B, A versus C, B versus C. When you compare, when you see in any study, investigator is assigned alpha value of 0.05, not more than that. So in a situation where you have three groups, so A versus B, B versus C, or A versus C. So when you do three comparisons, you can't take 0.05, 0.05, 0.05. That makes 0.15, which is more than 0.05. So therefore, you have to spend out of what you have. So do you do Bonferroni correction, and first one, it has to be 0.016, second has to be, you modify your p value for individual comparison, 0.016, 0.016, right? So this problem is more common in genetic studies. When they do it there, they say that I have these are the ones which are known genes, and these are the ones I have, these are the ones which are novel or whatever, right? So when they do all of this, they have their p value has to be modified. If they don't modify, then they say this is the gene, so that is false discovery or false positive.
Thank you, sir. Thank you. Yes, so I think we'll stop with this for this session. Thank you so much, sir, once again. Your knowledge of biostatistics and the way you have explained it, such a vast topic. It's really an ocean in a short time. It's really eye-opener for many of us. Thank you so much. We're looking forward to having you in future workshops also
Sure,
that we do in SRM. Thank you so much, sir.
Thank you. Thank you.
We move on to the next session. Dr. Stanley will be taking a session on bibliography. When we write a protocol, the last part of it is to have a good bibliography. And of course, when we even do a thesis or a journal manuscript, how exactly to go about managing your references. Dr. Stanley is, of course, my former colleague in JIPMA, and I would like Ms. Kavya to formally introduce Stanley.
Good evening, everyone. Good evening, sir. I'm really happy to introduce Dr. Xavier, sir. Excuse me. So Dr. Alfin Stanley Xavier, sir, he is currently working as an assistant professor from Department of Pharmacology, SRM CNRI, which is known as Sri Ramchandra Medical College, and as also an assistant physician in clinical pharmacology in Ramchandra Medical College, Porur. He is also a member of National Academy of Medical Sciences, NAMS India. He's a life member of Indian Pharmacological Society, IPS, the official body of Pharmacological Society in India. He is a life member of Indian Society of Rational Pharmacology and Therapeutics. His administrative contributions include clinical pharmacologist and medication safety coordinator, Ramchandra Medical College and Research Institute. He is also a department in charge, clinical pharmacy, SRMC, administration coordinators for drugs and poisons information center, and he is also a member of academic council, Srihar. He is also a resource person for academic programs and organized various workshops and conferences. He has 23 publications with around 260 citations. He has contributed 2 book chapters. He is also a reviewer for the scientific journals and also received Professor Manjeet Singh Prize for the research work in molecular biology at the 46th Annual Conference of Indian Pharmacological Society, which is held at the Bangalore during the year 2013, December 16 to 18. It's a pleasure to have you here with us, sir.
Thank you, ma'am. Thank you for the introduction.
So the session is over to you, sir.
Yes, ma'am. I will share my screen. Whether my screen is visible, ma'am?
Yes, sir.
Shall I start?
Yes, sir. Please.
Okay. Good afternoon, all. So after this 2 days session, hopefully this is the very much lighter session for your mind. So today in this session, I'm going to discuss about bibliography and referencing tools you will be seeing, and a demo about a referencing tool known as Zotero. Okay. So you may be preparing your manuscript, or you have written your protocol with a review of literature. So you might have asked to write the reference resources for your writer. Okay. So either you can do it manually. So if it is for a short text or only 4 or 5 references, you can do it manually. But if it is a large manuscript or a written dissertation, then there will be around 100 or 120 references. So it will be difficult to do manually. So this session, I will tell what is this bibliography and referencing, what are the different styles. Then we will see the demo about a software known as Zotero. So a reference is a list of resources you have used when you write your review of literature or your write-up for the manuscript or your dissertation. So this provides information so that other people who are reading your material, they know that from where you have taken or from where you have taken this information, or they can go and read on that further. So it is, you are supporting your statements you are making in your write-up and claims in your own assignments. Okay. And also it will give idea about what are the related works which happened already in the field. So that is the main purpose of citing the references. So it is also as a giving credit with citation to the source of information who have used that one. And also it should be always be accurate so that your readers to trace the source of information you have used. And also even in not only as a manuscript in your protocol or a review of literature. So whoever is assessing that protocol, so if you are writing a grant request to agencies, so they may be looking to the related works which have been already have done in the field. So what are the different resources? Very common resources you will be citing electronic journals, e-journals and books, and online resources like web pages, websites, blogs, and conference papers, proceedings. So these are the things you will be the most common resources you want to cite in your write-up. So when to use reference? When you are using thoughts and ideas or any facts from a particular article, or when you're directly quoting that, or when you're taking the idea which they have projecting in their conclusion. So that situations you can cite the references from where you are taking the information. So there are two terms like bibliography and references. So bibliography is sort of a broad term. So it is any list of references. At the end of your write-up, you will be enlisting all the resources you have referred. Okay. So enlisting that references, whether you are citing those references inside your text or not, irrespective of that, that can be called as bibliography. For example, you are writing a 2-page review of literature write-up. At the end, you can list all the references. So that can be called as bibliography. But when you say references, where what your meaning is, you are citing the places where you are, like you are making a statement like vitamin D deficiency is almost 80 to 90% of the Indians have vitamin D deficiency. So near the line, you have to do an in-text citation. So either you can put a number, or either it can be you can mention the author name with the published year near the statement. So that is called as in-text citation. So only that cited works can be enlisted in the references. So that can be called as references. So references have in-text citation, and at the end of write-up, you are enlisting all the resources. Bibliography, it may or may not have a citation within the text. So there are many reference styles available. The two most common referencing styles are Vancouver and Harvard. So what is this referencing style is, in what way you are mentioning the in-text citation, and the when you are enlisting, what is the style you are following to write the resource. So that is called as referencing style. So in-text citations either can be as a number. So you can near the text, you can put it as a number 1, 2, 3, like the references you have put. And in the enlisting the references, according to the number, you will write down all the references you have added. Then reference list entries is how you are writing a, or how you are mentioning a source. For example, if you are writing a, like, okay. So in-text citation can be like this. So here they have written a statement. So within brackets, they have written the author last name and the published year as a reference. This is one way of in-text citation. In APA format and in Harvard format, they will be mentioning the in-text citation like this. Or you can put numbers. So here in this statement, the references 1, 6, 11 are the resources from where they have taken the information. So that way also it can be written. Then when you are taking and enlisting those references, there are different types. Here they are writing the author's name first with their last name and first and middle name as initials. Then the title of the paper, then the journal name, year, month, and issue number and pages. So this is one way of writing the references. So in Vancouver, we'll be writing in this format. So like that, if you are in text, in text, if you are mentioning as 1, 2, 3, so finally enlisting, they will be writing the references in the same order. In Harvard type and all, in text, they will be mentioning the author's name and their published year. And in the referencing, when you are enlisting, according to the alphabetical order of the last name, they will enlist all the references. Okay. So these are the two parts of citation and referencing. So for each source, there are some format which can be followed. So I am showing the common NLM type and Vancouver type. For example, if you are citing a website, then you can mention that source in this format, like it has author name, what is the title, then type of medium, it's the internet, then the place of publication and the publisher, and when it was last updated, and when it was cited, and what is the website from where it is available from. So this information should be in this order when you want to cite a website. So if you want to mention a conference proceedings from where you have taken some information, so it can be like this: the editors of the proceedings, then the conference title, the date of conference, then place and place of publication, publisher, and date of publication. So these information should be written in this order to cite a conference proceeding. So like that for dissertation or to mention a patent. So for each format, the Vancouver Guide or Harvard, they have given a format which we can follow to cite the resource. So coming to reference managers, so if you are writing it's a brief text with 4 or 5 references, then you can do it manually. But if you have a large write-up having more than 100 references or more than 30, 40 references, definitely you need a tool to insert these references in your write-up. Okay. So these referencing managers, they are helpful to insert citation and insert the bibliography at the end of write-up. And if the journal editor is suggesting to use a different style, you can easily switch between different referencing styles using these tools. So there are common referencing tools like the Zotero, Mendeley, EndNote, Reference Manager, and others, RefWorks and all. So in that, Zotero and Mendeley are very commonly used. It is freely available. So EndNote is also having a basic version which can be freely used, but you need a institutional login. Okay. So since this first Zotero and Mendeley it will be easier for the beginners, so I will try to give a demo on Zotero. So comparing each referencing tools, each have its own advantage and service, but most of the formats will be same. Basically, you are trying to import the information from a website or from an article to the software, so that if you have 30 articles, each of thing you will insert in that. Then in your Word, where you have typed, you are going to insert the citation from the software. Then at the end, you are going to enlist this bibliography. That's the basic things you are going to do when you are using a reference managing tool. So I have sent you a installation recommendation, means how to install. So for saving time, I have already installed the Zotero in my system. So for Zotero, it has a software for Windows, Mac, and Linux. So according to your thing, you can download the software. And also, you need a Zotero connector to connect the software to the browser you are using. Okay. So for we have for connector for Chrome, for my Firefox and Safari. You can, according to the browser you are using, you can download and install the connector. Then another thing is, the plugin for the software has to be installed for the Microsoft Word where you are typing. Am I audible now?
Yes, you're audible.
So in the Zotero version 5.0, so it has Word plugin within that bundle. So no need to separately install that. But some new versions of MS Word may have some problems in getting the plugin. So that you can just disable your antivirus program, and if you reinstall it, then you can get the plugin. Okay. For Mendeley also it has a software and a web importer and citation plugin for Word. So separately you can download and install. So more or less the importing and citing will be almost same for Zotero and Mendeley. Okay. So with this I am going to the demo. So whether you can you see the screen, a complete screen?
Yeah, we are able to see
the Word document, right?
Yeah, the Word document we're able to see.
So once you install the Zotero, it has a shortcut in the desktop. So you can just open that. So this is the home page of Zotero, where it has options, and in this middle pane where you can import the citations. Okay, so here it has icons. So to manually insert, or from the from identifiers, you can like PubMed ID or digital identification DOI. So from there also you can directly import here. So here in my library you can create a folder for this. So now I have created a folder for this demo. So now I have prepared a write-up, and while preparing the write-up, I have mentioned the authors and published here for my easy reference. Okay, and also I have saved the journal articles in a folder so that it will be easy for me to import those references and insert in the Word document. Okay. So now my objective is here I have to insert the particular article. Then finally all these references I have to enter under references. Okay. You have to enlist as a bibliography. Okay, as a first part, I have to open my browser. So from there, I'm going to search the articles and I'm going to import those articles inside my software. So now I have opened the Google Chrome browser. So here you can see under extensions, I have Zotero as an extension. So once you install the software, you have to install also the connector to the browser. So after installing that, you will get an icon here as a add-on. Okay. So now the first article to cite is Bhattacharya 2015. So in the browser now I am searching for the article. So now I have opened the article in the Chrome. So here you can see once you open the page of the article, here you can see the icon as a, so it is with the dotted lines inside a box. So it's showing that it is a journal article. Okay. So from this, I should have already opened the folder in the Zotero. So from the browser, if I click on this, it will directly import to the demo folder of Zotero. Okay. So if you go and see here, it has imported the journal information inside the Zomato pane, in the Zotero pane. So coming to the next article, so now I want to include Lakshmi C 2007. So I'm just again searching for the article in the browser. So I have opened the same article in the PubMed. So here you can see that it is showing access to the article information for Zotero. So I can directly import this to the folder. So there is other way also to, like, I can copy this PubMed ID. So here there is an option like add item by identifier. So if you click here, so you can enter the DOI, ISBN or PMID number. So I am inserting the PMID and enter. So here again it is imported. So these are different ways you can import the citation from the browser, or through the PMID or DOI number also you can do. Okay, so since it is duplicated, I am removing one. Then third article, I have to insert Sharma VK 2012. So I have saved this. So now I have seen the icon here. So I am importing this to the Zotero. So now we have imported 3 articles. So now, so these are the articles which you are importing. So what if you want to import a book reference? So here I have written something about the immunosuppressants, so which I have taken from the Goodman-Gilman 13th edition under the immunosuppressants. If the book has a e-book version, then it will be easier for us. For example, I want to import from the Goodman and Gilman. So it has a e-book version. Okay. So I am just going to the so it has sections, and I want to cite the topic on immunosuppressants. So I'm just clicking on that. So now here you can see now this icon has changed as an open book. So this means it is a chapter from a book you are going to cite this. Okay. If it is a closed book, then you are citing the complete book. Okay. So once I open this thing in the browser, it will show it as a open book. So I have selected the chapter from where I have taken the information. Now I want to import this. Okay, so just I am clicking on that. So it is getting saved under demo. So you can go back to Zotero. So here you can see immunosuppressants and tolerogens chapter from the Goodman and Gilman has been imported. So what if the book is not available in e-format? Okay. So in that case, you can download the book information from Amazon also you can download like. So here you can see the icon as a closed book. So this is a to import citation for the book. Okay. So if you have a ebook format in the website, it will be easy to directly import the chapter information. So if it is a closed book, then also you can import, but after some time you have to, means after you add the bibliography, some of the information you have to edit after that. Okay. So this also I am importing. So here you can see the chapter version shows the chapter and the authors of the chapter, and the book version shows the title of the book with the editor's name. Okay. So now I have imported all these things. So in the text, if I want to, how to insert that citation. Okay. So now in this place, I want to insert the citation. Just so I'm going to Zotero inside the word. So here add citation, add or edit bibliography, then refresh, unlink citations and document preferences are there. So document preferences, which style I want to introduce, so that I can select. So here already there are styles which are enlisted here: American Medical Association, APA, Harvard, Chicago Manual of Style, and Nature, Vancouver. So I want to introduce in the Vancouver format. So first I can select the style. If your style which you want is not present here, so that also you can import inside the software. Okay. So now it is present, so I am selecting that. Now I am adding a citation. So it is showing a pane like this. So then it has two views. I can go to classic view. So I can see whatever I have imported inside the Zotero. So here I want to introduce the Bhattaria et al. 2015. So I am clicking on that. So it has been imported. So in-text citation is in numbers and it is in superscript with square brackets. So I have imported. Now here I want to import Lakshmi 2007. So again, add citation, classic view. So Lakshmi, so that I am clicking and I am importing here. So Sharma VK, add citation, classic view, and whatever I have imported, I am pressing, so importing that. And here I want to, in the last, here I want to introduce the citation from Goodman and Gilman. So again, add citation, classic view. So immunosuppressants chapter, I am clicking. So now I have introduced the in-text citations. Here you can see 1, 2, 3, and 4. Now how to add the bibliography? So under references, if you want to introduce bibliography, now add bibliography. So this will automatically add the information for your bibliography. Okay, see how simple it is. Now, in case if the journal editor or your guide who wants the referencing style in some other format, not in the Vancouver. So now you can go to document preferences. So they want in Harvard style. Okay. So you can click on the style. So now here you can see. So this author name and year, this is a Harvard in-text citation type. And here also you can see according to alphabetical order of the last name, the references are being enlisted. Okay. So easily you can switch between the styles. Okay. That is one advantage here. Then if you want to edit something, that also can be done. So if you want to edit some of the references in the bibliography, that can be done. And once you edit, means at the last step only you have to edit if you want to do some minor corrections in the thing. So after that, if you refresh, automatically it will, according to the document preference you have selected, it will change. Okay. So always edit only at the end. So now again I want to move to different document style like Vancouver, not with superscript, just a Vancouver I want to select. So now again it is with the normal brackets, not superscript, and the bibliography also has been changed. Okay. Now if you want to introduce some new referencing style, So how you can introduce? So you can go to manage styles. So here you can see there are different options there in Zotero preferences. Get additional styles. Okay. So here you can click. So whatever styles you want. Okay. So they have almost 9,887 styles of referencing. So you can search in this, or if you know by name. Okay. So for example, International Journal of Emergency Medicine. So they have, so all these styles, they have only minor variations. So the purpose of having a unique style for their journalist to have a uniformity so that the standard will be maintained. But main purpose is to cite the resources what you have from where you have gathered the information. So that is the main purpose. Only the citation styles will be changing. If you keep the cursor on each citation style, there you can see that how the in-text citation will be and how the bibliography will be. So sometimes they may be asking you a style which may not be there in this, but you can just look at this each style and which is very much closer to what style they are asking, that you can import from here. Like I want to import International Journal of Food Microbiology. So I am now here you can see, so that the additional styles have been downloaded to the style manager. Okay. So now if the document preferences I am selecting International Journal of Food Microbiology. So now automatically the referencing are getting changed. Here you can see from Vancouver it has changed to the newly downloaded citation style. So this is how it can be easily done. So apart from that, if you want to work in different systems, okay, so you can have a username login in Zotero. You can log in there, and you can sync your information which you are doing in a particular system to the Zotero website. So when you switch to the some other device, from there you can easily sync to your account. So here in document preferences, here there's an option called a sync. Okay. So once you create a Zotero username password, so you can enter the username password here and you can sync with your account. So in any device, if you open the reference manager, you can see all the citations you have imported, and from there you can insert those citations to the Word document. Okay, thank you. Almost I have finished. If you have any clarifications in using this tool, please ask so I can answer.
So, Dr. Stanley, there are a few questions.
Yes, sir.
I'll just read those questions for you.
Okay.
One is how to add a single reference in between during editing that cannot be saved in Zotero.
Okay, sir. Means they want to manually edit it.
Yes, I think probably a reference cannot be, Zotero is not able to retrieve. So manually you enter the reference in Zotero and then add that inside.
Okay,
So
that can
basically
be.
manually, yeah.
Okay, so in the Zotero pane here, you can see the option as new item. So where you can, like if I want to select an article, so I am just clicking. So here this.
Can you mute your audios, please? Please mute your audios.
Okay. So this importing, instead of from the browser, I can do it manually also. So like I'm clicking on the first option, add item. So what type of item is this. If it is a journal article, okay, I'm journal article, I'm clicking. See now, means you can't able to find the article in the browser, but you know the hard copy, you have the hard copy. So you can type all the information, title, what is the author's name, publication, volume. So once you insert this, this can be added in the citation panel. So from there you can easily insert. So that is how it can be done manually.
Okay, difference between cross reference versus cross citation.
Means they are asking about the tool or
Not the tool, I think. I think generally he's asking.
No, I can't understand what they are asking, sir. Cross citation and cross referencing.
Okay, I think we will leave that. Is there any Chrome extension for Mendeley similar to Zotero?
Yes, sir. So if you see in the Mendeley, so they have also a separate downloading software link, and also they have browser connectors also you can install, and also you have separate plugin for your word processor. So all these can be connected so that from browser you can download to Mendeley, then from Mendeley you can insert the citation in the word processor.
Okay, and one more question. Would it be possible to import these references to a Google Docs document?
It can be exported, sir, but not as a Google document. Like for example, here it has a export option where it has a RDF file format, and also like BibTeX format, which is commonly used, BibTeX, and RIS format. So these are some of the common formats which can interchangeably used between referencing tools. Like BibTeX format, I can export all the citations, or in RIS format. Then I can import those from the file. I can import those references in either Mendeley or in EndNote. So like that it can be done. So here it has options almost an RDF file, BibTeX, and RIS. So these are the common formats which are being used for exporting and importing.
Okay. 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 If any other questions related to, yeah, that's what. So the thing is, I think in what the question was in Google Docs, like just like how normal Microsoft Word, we have this converter. I mean connector. I think Google Docs doesn't have a connector, right?
No, sir. Only Word format, Microsoft, and another LibreWord. So those are the two word processes.
Yes.
Which have connectors to the Zotero and Mendeley.
Yeah, exactly. So I'm answering the question that it cannot be done basically.
Yes,
you need to use a normal Word document. Okay.
Yes, sir.
How to add a newspaper article or blog from internet, for example, Times of India, using Zotero manually or using extension?
You can browser, you can go to the web page. So from web page in the browser, you can directly import like how you are doing for an article.
Okay.
So if we open a web page, here you can see this icon. This is a web page icon.
Okay.
So from Times of India, particular article, that same page, from there you can import to the Zotero and you can cite it.
Okay. Okay. I think that's the end of this session. So thank you. Thank you, Stanley. So I think it was a very useful presentation for all of us, especially handling references when you have too many. It's always a challenge. So using these softwares which are freely available is a big bonus. It will help us to do our work much faster and avoid the hassle of managing too many references. So we'll just go for a few questions that are asked at the beginning during the sessions. We'll try to answer some of them. So Dr. Satyajit, are you here? Okay. Fine. So morning, there were a few questions, some of which I'll take. In the interest of time, we'll keep it short. So question was on a difference between intention to treat analysis versus per protocol analysis. So the difference between the two is that in an intention to treat analysis, you take patients who are randomized into the trial take their data irrespective of whether they have completed the complete duration of the trial. Even if they have taken only a few doses, you continue taking them. You do not leave those participants. Whereas in a per protocol analysis, you take the data of only people who have completed the protocol, who have, in whom you have the complete data. In such patients, you do an analysis, then it becomes a per protocol analysis. So in these days, most of the international journals, top journals, they expect that the author does both the types of analysis, compares and sees if there is any difference in the outcome measures between the two analyses. And in case it is not there, then it has a stronger basis. The results have a much stronger basis. Which authority grants permission to perform placebo-administered study? So there is no specific authority for placebo-administered study. Usually, placebo-administered studies are heavily used in regulatory trials, but of course, it can be done even in academic trials. So essentially, the same ethics committee, local ethics committee, as well as the regulatory, that is CDSO approval for regulatory trials would apply. What can be a placebo drug? Is there any certification that selected placebos completely inert? There are there are pharmaceutical companies which are into the business of manufacturing placebo tablets. So once they provide that, they'll be giving a certificate stating what is the composition, something like lactose or some inert substance which is not having any pharmacological activity. And does placebo administered research, can it be done in university or it can be done by individual researcher? So that's what, there is nothing like specific individuals only are authorized to use placebo. As long as it is having an ethical usage, the protocol is using placebos in an ethical fashion as recommended by the ICMR guidelines as well as New Drugs and Clinical Trials Rules 2019. It is acceptable to use placebo irrespective of the context. So we are coming to the end of this workshop. We once again want to thank all the participants for having actively participated in the session. Madam, are you there, ma'am?
Yes, so only few.
Yes, ma'am, please.
Good afternoon, everybody. Audible? Is it audible?
Yes, ma'am. Yes, ma'am.
Good afternoon, everybody. I thank the speakers, Dr. Smita, Dr. Paranivel, Dr. Melvin Judge, Dr. Satyajit, Dr. Pandey, Dr. Stanley for your valuable speech, which is presented in an easy way and it will be useful for the participants. Finally, I thank the participants for their very good participation in the many questions have been posted and it was very interesting. And especially I want to thank Dr. Melvin and his team for his excellent work. Thank you.
Thank you, ma'am. Thank you. So, there is a feedback form that is provided in the chat box. I request all of you to click on it, fill the feedback form before leaving the meeting room. And as I told before, we will be providing some documents like how we provided yesterday. We'll be providing some documents for your further reading today, as well as the next 2-3 days. So please take time to read those documents, go through those articles, because we are going to give you some assignments based on those articles. Only if you have read those articles, you will be able to fill those assignments and complete those assignments. So please, and we will also send you the link of this 2 days sessions. It will be uploaded in Google Drive and the link will be sent to you. So in case you have missed any one session, you can even go through that session thoroughly and make the best use of it. And we understand that some of the sessions might have been difficult because it was a very short time given for each session. So hopefully in future we'll have a more detailed workshop on some of these topics if as the time goes. So at this time, we also wish to thank Dr. Jamna Rani, HOD, Department of Pharmacology and Clinical Pharmacology, for helping us and guiding us and facilitating us throughout this workshop. And also all the speakers, Dr. Satyajit, Dr. Smita Patnaik, Dr. Ravindra, RM Pandey, Dr. Stanley, and every other speaker who was with us the last 2 days. We wish to thank them for giving their time, Dr. Palanivel too. So, and special thanks to the organizing committee members, Ms. Kavya, Ms. Indra, for taking care of all the arrangements the last 1 week. Also, I mean, a lot of activities required, a lot of coordination with the delegates and the speakers. So we thank Ms. Kavya and Ms. Indra for helping in all these coordinating activities. And thank you, delegates. You have come from, I mean, you have, even though you are at your place, but from different parts of the country, we are able to connect through this Zoom platform. So it's a wonderful opportunity to connect. So hopefully we will continue interacting through the assignments and the interactions that we have with you. And based on the assignments, you will be provided the certificates. So please take the assignments very seriously. We hope that even the assignment will be a learning exercise for you. So with that note, I wish to conclude this workshop. Looking forward to meet you sometime, maybe physically also in future as the pandemic subsides. Thank God for helping everything go on successfully. And I hope this workshop will have a significant impact in your career as a researcher. God bless you.